The cardiovascular system. This happens to be one of my favorites. The heart is actually my favorite organ just because it's super cool. And there's so many things that it does. So this is going to be one of those chapters where it starts out fairly easy, and then we're going to get into some pretty challenging stuff in terms of some of the physiology that happens with how the ions function and how the heart is able to be stimulated. Not only do you have muscle, which is what you see here, but we also have a conduction system, and we have the sympathetic and parasympathetic. So we're going to go through and talk about how it is that all that works together. So what you're seeing here, these are the layers of the heart. And the outermost layer of the heart is called the epicardium, or is also known as the visceral pericardium. So the outermost layer that you have in your pericardial sac, which is the membrane that surrounds the heart, you have the parietal pericardium and then the visceral pericardium. And the little space in between, which is right here, this is the pericardial cavity. And this is where sometimes you will have some effusion that happens of fluid that can come from the heart, or if it's blood, but it's fluid that will get stuck in between the two membranes, and then what that can cause is what's called cardiac tamponade, and that will cause some serious problems in terms of the ability for the heart to function properly. Then the middle layer of the heart is called the myocardium, and this is the most important layer, obviously, because this is where the muscle is and what allows our heart to contract and do what it does. And you'll see that the word myo, this means muscle, and then cardi means heart. Then the endocardium, this is the innermost layer, and this layer is the inner lining. And what's really important about this lining is that it's actually kind of slimy, so that the blood does not end up getting kind of stuck or clotted up against the actual wall of the heart. And then you'll notice that it's got a little bit of kind of grooves and stuff on the inside. And what this is called trabeculate carne, and what it does is that it allows for the blood, when the heart contracts, to increase its turbulence so that it can be ejected of the blood out more forcefully and more efficiently. So it's kind of like having rapids inside your heart. It allows for that blood to flow more quickly. Then the chambers of the heart, we have 4, and the 2 upper ones are called the atria. The 2 bottom ones are called the ventricles. And they are each separated by an interatrial septum between the atria, and then the ventricles have the interventricular septum. So what you'll see in here, and you can't really see the interatrial septum, but there is one between the 2 atria that you see here. And then these 2 down here are the left and right ventricle. And then this right here is that interatrial septum. And why this matters is because this is what divides the 2 ventricles and basically separates the right side of the heart from the left, or as you see here in blue, in biology, blue on a vein or a vessel means that it's talking about that the blood there is deoxygenated. Here, the artery is red, and in this case, this artery happens to have oxygen. So where you see red vessels, you're going to notice that it's going to be oxygenated blood, and where you see blue vessels, that is going to be deoxygenated blood. So what I've done in this diagram is that you can see that the right atrium and the right ventricle are in blue, the left atrium and the left ventricle are in red. So that is telling you the type of blood that passes through these chambers. Then you'll notice over here that this shows the pattern of the ribbon of the actual heart muscle. So the 2 upper chambers, these are the atria, and you'll notice that they each kind of have a circular pattern to the muscle itself. And so what they do is when they contract, they actually push blood down, and they push it down through these valves into the ventricles. So the job of the atria is to actually top off the ventricles. Then you'll notice that the bottom part, where the ventricles are, the ribbon is shaped a little bit differently. It's actually at an apex, which is right here at the tip of the heart, and when the heart contracts, it actually kind of turns in a way and kind of twists like a washcloth, and then it ejects the blood out into its respective vessels. The 4 valves that are in the heart, the first 2 that are between the atria and the ventricles, on the right side, the tricuspid valve is between the right atrium and the right ventricle, and on the left, the bicuspid valve is between the left atrium and the left ventricle. The pulmonary valve is considered to be a semilunar valve, and it's between the right ventricle and the pulmonary trunk, and then the aortic valve is between the left ventricle and the aorta. So when you look at the images, you'll notice that tri-right, so tri-right is another way that I've had people remember this, and this is a cuspid valve. So what that means is that these little valves actually point down into the ventricle, as you can see here, and these pointy little cusps are actually attached to these little muscles here that are called papillary muscles. And what the papillary muscles do is that they actually contract when the actual ventricles contract. And what they do is they actually keep these valves closed so that the blood won't regurgitate back up into the atria when the ventricles contract. Then the semilunar valves are right here. So this is the pulmonary semilunar, and then on this side is the aortic semilunar valve. And what you're looking at here is that they're basically set up so that when the pressure of the blood pushes up against the valve this way, it opens and then the blood enters into the vessel. Same thing with right here with the actual aortic valve. Then just so you know, the arteries, the actual definition of an artery is a blood vessel that carries blood away from the heart. The farther they go from the heart, they get smaller and smaller and smaller, and they become arterioles until they enter into the capillary bed. And then what happens there is that the further it goes from the heart, that is where the arterial pressure is going to continue to decrease, and it's going to increase with what's called with increasing distance from the left ventricle. So as you get down into the smaller vessels, the actual pressure and the velocity of the blood flow decreases. When you look at the actual arteries that are coming off the aorta, and again, these are red, and these are arteries that you do need to know, and it's very important that you know these because what happens is that this is how your brain and the upper body and also the heart are also going to get their oxygenated blood. So you'll see that they're all in red. What you see here in blue, these are the right and the left pulmonary artery, and then this is the pulmonary trunk, which is the big vessel that divides into the left and to the right pulmonary artery. These are going to carry deoxygenated blood to the lungs to get oxygenated, and then these red pulmonary veins are what bring the blood back after oxygenating it from the lungs. So up here at the very top, this is called the aortic arch. And in here, you actually have baroreceptors that measure how much pressure is passing through the aorta. So this very top part that you see up here, this is the brachiocephalic trunk. And this brachiocephalic trunk divides into the right common carotid artery that then divides into the internal and external carotid arteries on the right, and it also divides into the right subclavian. When you see the word common or you see the word trunk, that usually means that that particular vessel is going to divide. Common usually means it's going to divide into a left and right. Then in the middle, this is the left common carotid artery. This will divide into the internal left carotid artery and the external left carotid artery. And then on the far left, over here, this is the left subclavian artery, and then this is going to feed in to the actual left side of the upper body. When you look at the back side, you can see a little bit better that the actual vena cava, so this is your superior, your inferior, this is the right atrium, and then these vessels, these are your pulmonary veins that return the blood back to the left side of the heart. And this is the left atrium. So from the back side again, here's your aorta, you've got your brachiocephalic trunk, the left common carotid in the middle, and then the left subclavian artery. When you look at the actual subdivision, and this is kind of like what I call the plumbing diagram. It's a thematic diagram of what you're going to see here on this vessel, but I just want to start with this one. You'll notice here that the aortic arch, so the blood's coming up and out off the aorta, and what you have here, here's the brachiocephalic trunk, here's the left common carotid, and then right here is the left subclavian. So what happens is that the actual subclavian arteries, what they do is they divide, and they have two little branches that are very, very important. This right subclavian and this left subclavian, this branch here and this branch right here, become what are called the right and left vertebral arteries. And why this matters is because these right and left vertebral arteries are actually passing through the posterior part in what's called the transverse process of the cervical vertebrae. And what that does is that it actually brings blood up into the back side of the brain, and it's going to feed into this very important artery, which is called the basilar artery. And this is how the brainstem actually gets its blood flow. Then this will branch off, and then it becomes the right posterior cerebral artery and the left posterior cerebral artery, and then those connect through these communicating arteries into the cerebral arterial circle, which is also known as the circle of Willis, which then branches off into the right middle cerebral artery to feed the underside and mid portion of the brain, and then this is going to be the left one. Then on the anterior portion, you're going to have the anterior communicating artery, and then you have the right and the left anterior cerebral arteries. So this is how the majority of the blood goes in to the brain, and it feeds off from your vertebral arteries that come in and feed this way, and then also through these guys, which are going to be the internal carotid arteries that are passing through. And then you'll note that the brachiocephalic trunk then will subdivide into obviously the internal and external carotid artery, but then it also feeds in, and then it feeds into the facial arteries that are then going to feed the different parts of the face, and then this one goes in to the thyroid gland. So it's very important, obviously, that these branches that are coming right off the aorta are clear, because they supply significant blood flow to the top of the portion up here that is going to be the underside of the brain. Then when you look at the actual schematic, here you can see where that brachiocephalic trunk divides into the common carotid artery, the right. So this is going to be the external branch, and then this is the internal branch that feeds in and then goes into this right side of the head. And then over here, you can see that this part of the subclavian branch, this is the right subclavian, then feeds in to what you see here, which is the right vertebral artery, which then passes through and then is going to feed into that posterior portion that gives rise to the blood that supplies the actual brainstem. And then also you'll note that it's blood that is going to end up supplying the posterior portion of the head. So very, very important that this blood flow is open. Then veins themselves, their definition is that they return blood to the heart. And the way they work is they come off the capillary beds, and they start very, very small and then increase in size the closer they get to the heart. And as the size goes up, the pressure also gradually increases the larger they get to the heart. So the vena cavas are going to be the vessels that have the largest pressure in terms of venous pressure and returning the blood back to the heart. So when you look at the image, here's the superior vena cava. It returns blood from the upper body and the brain. And then the inferior vena cava returns blood from below the diaphragm. Then the pulmonary veins, these are going to be red because they are returning oxygenated blood back into the left atrium. So when you look at the back side, here are the pulmonary veins returning the blood to the left atrium, and then here's the superior vena cava, and this is the inferior vena cava. Then another very important vessel that you see on the back is this guy right here. This is called the coronary sinus, and what this vessel does is that the blood that returns from the heart muscle itself after it was oxygenated and the tissue went through its metabolic process, you'll see that there are these veins that run parallel to where all the vessels in the heart are. And so you can see here, this is called the great cardiac vein. These are the smaller cardiac veins, and they run right along the major arteries of the heart that we're going to learn in a little bit. But this coronary sinus is what empties also into the right atrium, because again, it's deoxygenated blood. So this right side of the heart, on the upper chamber, the right atrium, empties the superior vena cava, inferior vena cava, and the coronary sinus. Then again, you can see here that we've got another thematic plumbing diagram in terms of the blood that is going to return back into that subclavian vein and then the superior vena cava. But first, on both the right and the left, we have these brachiocephalic veins. And what this means is that they are veins that are returning blood from the arm, upper body, and from the head, the brain. So when you look at this image, you'll note that there are these words sinuses on very large vessels. These are also the veins that are found in the brain that return the major portions of that blood flow. As it's gone through, it's been metabolized, oxygenated, drops off the oxygen, picks up the carbon dioxide, and then the waste product obviously enters into the veins. And then these veins are going to then return the blood back to the jugular veins, and this is your right internal jugular. Obviously, you have one on the left. And then, just like we had an external jugular vein here, these are the veins. This external jugular is what is going to be draining the veins that are going from the backside, like the occipital vein, and then the veins that are draining from the facial portion of the face. You can kind of see that here as well. Then what you see here is very similar to the one that had the arterial network on here. Here's that vertebral vein that then will drain into the right subclavian, and then that drains down into the brachiocephalic, and then that goes into the superior vena cava. So you can see that our brain, our head, very good blood supply. All right, so collectively, here they are all together, and you're not going to have a diagram that you're going to have to label or anything like that, but this just kind of helps you see where everything kind of works together and where the oxygenated vessels are and where the deoxygenated vessels are. And so once again, just to remember and kind of hopefully bring back a little memory here, your right atrium is going to be carrying deoxygenated blood. That has blood that's coming from the superior vena cava, the inferior vena cava, and the backside, the coronary sinus. Then your arterial network is going to be the pulmonary trunk. So on the anterior portion, here comes the pulmonary trunk. That's going to branch into the left and the right pulmonary arteries that carry deoxygenated blood to the lungs, and then the deoxygenated blood will return back to the heart through the left and right pulmonary veins that then return back to the left atrium. And then on the very top, the aortic arch is where the branch is, and it's going to contain the left subclavian artery, the left common carotid that branches into the internal and external left carotid artery, and then the brachiocephalic trunk, which is going to branch into the right subclavian and the right common carotid, and then the right common carotid will then branch into the internal and external carotid artery on the right side. So in red, here are the oxygenated blood vessels that you're going to have that are part of the heart anatomy, the right and left pulmonary veins, the aorta, and then the left subclavian, left common carotid, the brachiocephalic trunk that divides into the right common carotid and the right subclavian. Then your vessels again that carry the deoxygenated blood into the heart itself, the inferior vena cava, superior vena cava, the pulmonary trunk, that one subdivides into the left and right pulmonary arteries, and then don't forget the coronary sinus that returns back the blood from the heart itself. So what we have with our circulatory system is a closed system, which means we don't want any leaks. So when we have the heart here, this is where the aortic arch is, then that's going to go into the actual aorta. And the aorta and the pulmonary trunk are considered to be elastic arteries. I'm going to focus primarily here on the systemic circulation, which means it's outside of the heart. And what you're seeing is that these elastic arteries will then give branches to what's called the muscular arteries. And these muscular arteries distribute the blood to the primary organs. Then from there, they're going to get smaller. And when they get smaller, they become little arterioles. And so what happens is, up here at the top where you have one big artery, right, we've got the aorta, and obviously over on the pulmonary side, you've got the pulmonary trunk. These vessels are very thick. They have a very thick musculature in the middle layer, and they're able to withstand a lot of pressure. But as they get smaller and smaller and smaller, as they're going into these little capillary beds, which is what you see here, these little guys are going to be the larger determinant in terms of what is a person's diastolic pressure. And so what that means is that how easily is that blood able to flow. And so you want the blood to flow nice and easy when it gets down into these areas where we have all these capillary beds. And so you don't want there to be a lot of clogging down there, because if there is, it's going to make it more difficult for that blood to flow into the capillary beds. Then on the venous side of a capillary bed, that is where the blood will now go from being in a very small venule to a little bit larger vein, and then they call it capacitance, like for example, your femoral vein. And then they'll start getting bigger and bigger, and then they will feed into the vena cavas, and then they return back to the heart. And then what you're seeing here in green, this is that network that we talked about in the immune chapter. It's called the lymphatic vessels, and those always have a supply to the capillary beds to help remove the excess fluid. When you look at what an artery looks like compared to a vein, the biggest thing you'll note is that the musculature in the middle layer that is called the tunica media, which is right here, is much thicker on the arterial side than it is on the venous side. Both of these, all the vessels, are supplied specifically by the sympathetic nervous system. So the sympathetic branch of the autonomic nervous system is what supplies the innervation to this muscle. So when the sympathetic system kicks on, what it does is it causes constriction and increases pressure in both the arteries and in the veins. And that's a very important thing that you need to remember. The reason why we have blood that flows primarily into our gut and our kidneys when we're at rest is because the parasympathetic system, what it does is that it actually inhibits the sympathetic system when it's activated. But as soon as the sympathetic system kicks on, boom, it will always take over the parasympathetic system. So the layers that you see here, the tunica intima is the innermost layer of a vessel. This is where the endothelium is located, and this is also where, when there's damage, that there will be healing that will take place based on the blood clotting and what we talked about and how the body stops the bleed. This is also where, as you can imagine, where you get into the smaller arteries, where we start having any kind of plaque buildup that narrows the space. That also decreases blood flow through that area and increases the resistance, so it makes it harder for the blood to get through. Then our capillaries, they are set up. Obviously, you're going to have an arterial side, and then you're going to have a venous side, and then the blood's going to flow from the arterial side into the venous side. The precapillary sphincters, which you see here on this image, these little guys, what their job is to regulate blood flow into the actual capillary bed. And what you don't see on the image here is, remember, all around this little capillary bed, there are tissue cells. And in between the tissue cells and the vessel, there is a teeny tiny space that is called the interstitial space. And that is where you have a minimal amount of fluid that is called the interstitial fluid. So when the vessel is open, and these little precapillary sphincters are open, blood is going to flow into that capillary bed. How these open is dependent upon the metabolic activity of the actual tissue. So, for example, if you are in skeletal muscle or it's a digestive tract and you're in a digestive state, these vessels are going to be having blood come in because the tissue that it feeds is more metabolically active. So the metabolism and the metabolism byproducts, carbon dioxide, the water, the hydrogen ion that is produced as a result of the aerobic metabolism will cause these sphincters to open up. And then, once that metabolic pathway is done and the tissue basically decreases the active metabolic state, what happens is now the blood flow decreases through the tissue. So you still have some blood flow, but here in this image it's looking like it completely cut off the tissue. It doesn't do that. But it's going to significantly decrease how much blood is actually going into the capillary bed. And so what happens is that the blood actually shunts through and then travels onto the venous side without really dropping anything off at that tissue. What this does is that this is one of the reasons why we always have some saturation of oxygen even in venous blood, especially in the systemic circulation when it's returning back to the heart, because you never drop off 100% of the actual oxygen that you're carrying on that systemic arterial side. Then what our capillaries look like are three flavors. We have what's called continuous, fenestrated, and sinusoidal. The continuous capillaries, these are the ones that we have the most of. And you can see that there are little gaps in the actual vessel wall, but they're very, very small. And so you're not going to have a lot of stuff that actually passes through here. All right. And the red blood cells are going to stay in the actual vessel unless, of course, there's some kind of injury and then they start to leak. In the actual kidneys, we have a filtration system that is very important. These are actually called fenestrated capillaries. And what they are is they actually look like they have a little strainer where they allow more stuff to pass through. So for example, glucose, ions, these kinds of things are going to pass through into the capsule that is where the filtration system is located. So more bigger stuff is going to go through here. And then in your spleen, you'll note that the gaps in these capillary walls are much, much larger because the spleen and the liver are actually filtering a lot more than just our kidneys, and then obviously what happens in the continuous capillary beds that feed most of the tissues. So these sinusoidal capillaries, what they do is they allow more blood to enter into the actual liver and the spleen, and this is also why, I don't know if you like to eat liver, but this is why liver has that kind of metallic, bloody taste to it because of the significant amount of blood that goes in. Then what you see here is the actual just systemic circuit that happens, and then also the pulmonary circuit as it works with the heart. So the right side of the heart is called the pulmonary circuit, and it feeds the deoxygenated blood that returned from the systemic circulation and the coronary circulation into the pulmonary trunk, that then splits into the left and the right pulmonary veins, and they take the deoxygenated blood to the lungs. Then at the lungs, they will drop off the CO2, pick up the O2, and then the pulmonary veins will return the blood to the left side of the heart, as you see here, and that blood will then pass through into that left ventricle and then pump into the aorta and then get distributed into the systemic circulation. So pulmonary circuit involves the right heart. It includes the pulmonary trunk, the pulmonary arteries, the arterioles, and the alveolar capillaries that feed into the actual lungs. This is a short, low pressure circuit, and this is also why you'll note that the right side of the heart, the amount of muscle that's in that chamber is going to be much thicker or thinner than what you see on the left side, because the left side of the heart is the systemic circuit, and that one obviously is going to be much bigger. So left side pumps the oxygenated blood to the tissues, and that is known as the systemic circuit. So it goes into the aorta, the arteries, the arterioles, the capillaries. This is a long circuit, so the pressure is going to be much higher, and these systemic arteries are obviously carrying the oxygenated blood away from the heart to the tissues. And the reason why it's high pressure is because the heart muscle itself is much thicker, it can contract more forcefully, and the distance it has to go is much further. So your systemic circuit, this high pressure we're talking about, is actually your systolic blood pressure that you measure when we do a BP on the person's arm. So your systolic blood pressure is going to be that blood that exits from the heart during that left ventricular contraction. So this pressure here, the pressure is much greater, and the vessel itself and the distance that blood has to travel is much further. Over here, the right ventricle is much thinner than the left ventricle, and the blood supply is only going to the lungs. So it's a low pressure, lower volume, or not lower volume, but the volume is going to be very similar to what goes out into the systemic, but obviously it doesn't have to go as far. Then remember that your deoxygenated blood is going to return to the right atrium through the superior vena cava, inferior vena cava, and the coronary sinus. And then your oxygenated blood is going to go out into the aorta, and then it'll get distributed into the systemic circulation. The pulmonary venous blood will then return through the pulmonary veins into the left side of that heart. When you look at what happens as the blood returns to the heart, we have this thing that's called passive blood flow. And what that means is that the blood is passing from the right atrium into the right ventricle and from the left atrium into the left ventricle through their respective valves in a passive way, which means that the heart muscle is not contracting. Specifically, the atria are not contracting and they are relaxed. So in order for the ventricles to get what's called their end diastolic volume, the atria have to contract. So what you see here that is called the atrial kick, that is actually the atrial contraction, or what we call systole. The word systole means contract. And so what the atria do is, as the blood is returning back to the heart, they're going to be ready for those valves to open when the ventricles relax. When the valves open, that blood is going to flow passively into the actual ventricles. And then when the atria get the signal to contract from the cardiac conduction system, they contract and they top off the ventricles, and now you have your end diastolic volume. So when you look at this image, here is the blood returning from the superior and inferior vena cava and the coronary sinus, and on the left side you've got the blood returning to the left atrium. What I want you to understand is that the blood returns to the left and to the right at the exact same time. Most of the time when you learn blood flow through the heart, people are actually learning it is the order of deoxygenated blood as it enters the heart and then passes through and does its respective things going to the lungs. And we're going to go through that in a little bit. But it's very important that you understand that it isn't like the left side is waiting for the right side to get the blood to it, because it doesn't do that. If that happened, we would be passing out every other beat because we wouldn't be getting oxygen to the brain. And so that blood returns simultaneously to the left and the right side. Then once the blood has passively filled, and now we have the blood in the ventricles, what is going to happen next is we need these atria here to actually contract. And so when the atria contract, that is when we end up with the end diastolic volume, which is the maximal amount of blood that you're going to have in the ventricles prior to contraction. What's going to happen is as those ventricles fill and as we have the maximum volume of blood in there, the EDV, it actually creates pressure up against the valves, which aids in them closing so that there won't be a regurgitation up into the atria that they came from. But what will also happen is that the signal coming down through into the ventricles will then hit the little papillary muscles and cause them to pull on those little cords so that the actual valves stay shut. It's very important that that happen. Then once the ventricles contract, once they get their signal, now what happens is the ventricles will exert pressure against the blood will get ejected out of the heart, and the pressure of the blood as it pushes on the semilunar valves, the aortic and pulmonary, this is what happens. It opens up the valve, and now the blood goes into the actual vessels. So the blood will enter into the aorta up here and then into the pulmonary trunk that is going to go to the pulmonary arteries and then to the lungs. And then how they close is that the blood inside the actual vessels, the pulmonary trunk and the aorta, kind of sit back on the valves and close them. There are no little cords on these valves, nothing that's going to actually help in keeping them completely closed. And so what ends up happening is you need a little bit of back pressure so that those valves stay shut. All right, now what we have here is the actual ventricular contraction. So the ventricles are going to contract, and then remember that what has to happen is that the pressure generated in the actual ventricle, which is what we have in our, in our actual region down here, this pressure is really, really important because this is your ventricular systole. And so what has to occur is if the pressure here, down in the ventricles, is not strong enough to get the blood up through the aorta and through the pulmonary trunk, the blood would remain in here. So that is why our ventricles can create so much pressure to contract and then forcefully be able to eject the blood on out. So what has to happen is the right ventricle and the left ventricle have to exert the pressure strong enough and overcome what is called resistance, which means anything that is creating back pressure here in the pulmonary trunk or the aorta, or if something is going on with the valves themselves. If there's an issue on the valve where they're becoming stiffer, there's a blockage, something's going on, or there is too much thickening in the pulmonary trunk or arteries, or something's wrong in the aorta that's creating a lot of back pressure, then it's going to be much harder for those to open. So it's very important that the ventricles be able to exert the right amount of pressure to be able to open those semilunar valves and get the blood out. So they're always contracting against some resistance, but it's tolerable resistance. So once the ventricles contract and the blood is ejected out, we get this ESV, which is called end systolic volume. And what that means is that it's the amount of blood that is left behind in the ventricles after the ventricles have contracted. So once the ventricles contract, there is always going to be a little dribble of blood that's left behind here, and that is called the ESV. So if there is an issue with the ventricles where they're not contracting as forcefully, the amount of blood that is left behind, the end systolic volume, will actually be greater. And you don't want that because you're going to have blood coming back and passively filling the ventricles. So if you already have too much blood in the ventricles from the return, now you're going to have an issue where any blood that is coming in here, once it starts overflowing the ventricles, that blood will end up flowing back into the atria and then back to where they came from. So when you look at the actual systemic circuit, if you have too much ESV in the ventricles, so they didn't empty properly, as that blood is returning from the systemic circulation to the right and returning from the pulmonary circulation on the left, what's going to occur is if this side is still too full, then the backflow is going to end up going into systemic, and then it's going to cause edema, for example, in the legs. If it happens on the left, now the problem is the blood flow is going to flow back, and now you're going to have pulmonary edema in the lungs. So when you look at what is the blood flow from the right side of the heart to the left, all right, so when you learn blood flow through the heart, all right, this is normally what they teach. So we'll start on the right side. So the blood is going to return from the superior and inferior vena cava and the coronary sinus, and it empties into the right atrium. From there, it passes through the tricuspid valve, into the right ventricle, through the pulmonary semilunar valve, into the pulmonary trunk, to the right and left pulmonary arteries that take the blood away from the heart to the lungs. They will load the oxygen, unload the carbon dioxide, and then the right and left pulmonary veins will return oxygenated blood to the left atrium back to the heart. So once it gets into the left atrium, the oxygenated blood will pass through the bicuspid valve into the left ventricle, then from the left ventricle to the aortic valve, into the aorta, and then it will go into the coronary circulation, which is the heart, through the left and right coronary arteries, and then into the systemic circulation, which are the brachiocephalic trunk at the top of the arch, in the middle of the arch, the left common carotid, and then on the left of the arch, the left subclavian, and then it will descend through the descending aorta that goes through the thoracic cavity in the back, and then pass through the diaphragm and go into the abdomen where it becomes the abdominal aorta. So that is your pathway. When you see here is the actual coronary circulation. So it labels all the veins and all the actual arteries. For our purposes, you don't have to learn every single one of these arteries and every single one of these veins. We're just going to learn the main ones that are going to be related and correlate with what you're going to be learning in ACLS and with EKG. And so what this means is that it's very important that you start memorizing today what are these coronary arteries that matter so much. So when you look at how the heart receives its blood, at the base of the aortic valve, there are two little openings. The little teeny tiny openings that you see are these little guys right here. What they are they're going to feed into the right and to the left coronary artery. So these are the main, all right? This is how your heart gets blood to feed the coronary arteries that will then branch and feed the heart muscle. So you can imagine when there's an issue and there's a blockage in any of these openings, that is why a person usually dies from cardiac arrest, because that heart is not receiving any blood. This left side here that you're looking at, this is the left atrium. Over here, this is all part of the left atrium, so they kind of flapped it open. But this is where you have the actual opening of the little coronary arteries. So what happens is the blood at the base here of the aorta drips into, it literally dribbles in. This is your left coronary, this is your right coronary artery. And then the branches that you need to learn are that the left coronary artery branches into the circumflex, which is going to go around just underneath that left atrium and then goes back behind the heart and then will connect in the middle an anastomose, which means an anastomosis is a connection between two vessels, and then it connects to this posterior interventricular artery. Then the left coronary artery, the second branch it has, is called the left anterior descending, which is known as the LAD, and the LAD goes through the inferior portion down just to the side of the apex, which is this part, and then also connects posteriorly to that posterior interventricular artery. Then what you see here with the right coronary artery is going to branch into the right marginal artery, and then the right marginal artery primarily is going to be feeding that inferior portion of that left ventricle. There are little branches that are also going to be feeding the anterior portion of the actual right ventricle, but the big ones that we care about are the arteries that you see pictured here. And why we care about that is because where is it that they go? All right, so remember, the right coronary artery, the one you need to remember is that it branches along the side of that right ventricle, and it's called the right marginal. The left coronary rapidly divides into the left anterior descending, which is going to pass down and feed into the interventricular branches that are going to feed into the septum, which is very important, and also part of the left side of the heart. And then the circumflex is going to go around the left, just under that left atrium, and then go towards the posterior. So when you look at why we care that these vessels are open, is because it's directly related to what you're going to be learning about EKGs. And so the leads in a 12 lead, what they do is they tell you what part of the muscle of the heart, looking at it from 3 dimensions, what is going on. And so primarily what you're looking at when you see the EKG is you're looking for what's called a STEMI, right? Or some kind of irregularity. What I picked out here for this actual image is what we call a STEMI. So what you see here, this is an ST elevation. This right here, this little pattern, is indicative of what you would see in somebody who is having an MI, a myocardial infarction. An infarct means that blood flow in a vessel has cut off basically the oxygen to the tissue, and now the muscle is infarcting, which means it is dying. And so what you see here is, where is it happening? So where you see this, right, this ST elevation, it's going to tell you, oh, it's in this part of the heart. So for example, the leads you'll see are colored very similarly based on the areas that they're looking at for the heart. So lead 1 in AVL lead into, or they tell you about the high lateral wall of the left ventricle, which includes the circumflex artery, and also the AVL is that high lateral wall of the left ventricle. Then V5 and V6 look at the low lateral wall of the left ventricle, also looking at what is taking place in that circumflex artery. So if you saw some kind of ST elevation in 1 AVL and V5 or V6, then that would tell you, hey, something's going on. So you look up here, and here's 1 in AVL. There's no ST elevation here, but what you do see is a little dip right here. And that's something that needs to pique your curiosity, because look, it's also right here and definitely right here in that V6. And so there's something going on that should not be happening. Then what you look at is, okay, this 2-3 AVF over here, this is a right coronary artery, and that's 2, 3, and then AVF right here. And you note that 2 and 3 also has a little bit of funky, and then the AVF definitely has some funky. So what this is telling you is that what you see in this 2-3 AVF, something's going on, and in 1 AVL and V5, V6, something's going on, but they're not ST elevations. What they're showing you is what's called a reciprocal view, which means that the pattern that you're seeing here is going to be kind of opposite of what you see in the leads that would be basically mirror images, right, on the top side or the underside. So when you look at your V1, V2, V3, and V4, now let's take a look. V1 and V2 are going to give you information about the left anterior descending, and that feeds directly into what's called the interventricular septum. This is that thick muscle that is between the left and the right ventricle. And why we care about this is because through here, the left anterior descending is going to be feeding a lot of that left ventricle, and it's also where the cardiac conduction system is taking the message down to the bottom of the ventricles so that they will contract and be able to provide the blood for the systemic and pulmonary circulation. So look at this, V1, V2, we got a problem here. ST elevation, and then we look at V3 and V4, this is also LAD, and now you're definitely looking at that anterior wall of the left ventricle. So that whole anterior wall and the interventricular septum of the actual left ventricle is having a problem getting a blood supply. And so what this is indicating is that the LAD has a blockage. So when we look at the actual image, you can see what these leads are looking at. So here's your left coronary artery. Remember that they branch into the circumflex and then the LAD, and then the right coronary artery is going to branch into that right marginal. All right, so the circumflex artery is going to tell you that 1 AVL is what this area is going to be looking at. And so this would be tied directly to what is the circumflex artery doing. So if you saw ST elevation in 1 AVL or V5, V6, or these two together and not these, when they're in it, what's called a contiguous lead, 1 AVL is a contiguous lead, V5, V6 is a contiguous lead, that means that it is happening. If you just have ST elevation in 1 lead, doesn't mean that there's a MI going on there. All right, it has to be in 2 contiguous leads. Here's your LAD. So what we just saw back here, this ST elevation, right? V1, V2, V3, V4. So now we go back to the image, V1, V2, so that's the interventricular septum that passes in between the 2 ventricles, and V3, V4, the anterior wall of the left ventricle, that's this right here. Okay, so all of this right here is having an issue in terms of receiving blood. Then why you saw the little dip in 2,3-AVF and also in 1-AVL is that we're mainly looking here at the anterior wall. So it would be a mirror image. Like, think of it if you're standing on the edge of a really pretty pond. It's a perfectly clear day and you've got a mirror image down that you can look at that is reflecting what's above. That's essentially what the 2,3-AVF does with the 1-AVL and V5, V6. So that's essentially why we call it a reciprocal lead. Then the cardiac veins, the only one I really want you to know is the coronary sinus, which is what feeds into the right ventricle. The vein that runs right down the middle, parallel to that LAD, is called the great cardiac vein. In the posterior, the backside, it's called the middle cardiac vein. That one runs alongside, next to the posterior interventricular artery. And then you've got the anterior little cardiac veins that run down the right ventricle. And then the small cardiac vein, this one runs down the right marginal artery. But again, the only vein that I want you to know of the heart is right here, the coronary sinus. And then here's another really good image of what you see with the coronary sinus. All right, then in terms of what does a heart cell do and what does it look like? So you'll note here that the heart cell has actual striations. And why this matters is because you're looking at the pattern of the contractile proteins. And our skeletal muscle looks a lot like this, except that it doesn't have these intercalated discs. So the intercalated discs, what they do is they actually tell you the length of one cardiac cell. So from here to here is one cell, from here to here is another cell, from here to here, and it's just how the slice got done when they were looking at the tissue. But these are your cardiac cells themselves. And remember that the unique thing about cardiac muscle is that they have these intercalated discs. What we have in between the cells along the lengthwise, so this lengthwise portion in between here, are these things that are called gap junctions. And why these matter is because these are little channels that actually are allowing ions to pass through. So when we want the heart to depolarize, these little gap junctions actually make it easier for the heart muscle to depolarize. And then when we want it to repolarize, then the heart muscle is able to repolarize easily because of the setup of how the gap junctions are set up. So skeletal muscle doesn't have this, but the cardiac muscle does, because you want it to contract in what's called a functional syncytium, which means it's going in a very rhythmic pattern. When we zoom in and we actually look at the individual cardiac cell, they don't have the intercalated discs pictured here very well. They just kind of have them here. But what you're looking at when you zoom in microscopically into the cell, you'll notice that there are these little kind of Z lines here and here. And what they do is they actually are showing you the border of what's called a sarcomere. So from 1Z to 1Z, this right here is a sarcomere. These little stripes are where you see these darker lines and then the light blue lines. This is the light band, and then this is the dark band that you saw on the picture before. So where it looks darker, that is where the protein is that's darker, and then where it looks lighter, that is where the protein is that's lighter. So here's your dark bands, and then here are your little light areas. What you have in here is we have some microscopic stuff that is very important in terms of our heart muscle. And we also have this stuff in our skeletal muscle. But in here, in the heart, this blue stuff is called sarcoplasmic reticulum. This is where it stores the within the cell, calcium. So the intracalcium storage, the intramuscular storage for the calcium, is in here in the sarcoplasmic reticulum. And you'll notice that there's this little tube in between bisecting the sarcoplasmic reticulum. This is called a T-tubule. And so you guys remember when you learned about the neurons and how they depolarize, right? Or how they can depolarize other cells. So what happens is that your heart cells are going to also be going through that depolarization action potential process. What happens with these little T-tubules is that they are open to the surface of the cell. So all along the surface of the cardiac cell, we also have ion channels. We have sodium channels, potassium channels, and stuff. All through here, inside the T-tubule, it's also lined with sodium channels and potassium channels. And why that matters is that when the signal is going and the action potential is generated on the actual cardiac cells, as the signal, the action potential is traveling on the surface of that cell, it will also send the signal down the T tubule. And then the sodium channels will open, boom, 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 boom. And then when that happens, it actually triggers the opening of the calcium channels that then allow calcium to go into the areas of the sarcomere. And why this matters is because you want the heart to contract. So when that calcium enters from the sarcoplasmic reticulum, triggered by the sodium gates that were opening as they were passing through the T tubule, then that calcium is going to go into here, into the sarcomere, and it's going to go find a very specific protein that is sitting on actin. So before I go there, I just want to kind of zoom in on here on the actual sarcomere and describe it a little bit better. So a Z disc to a Z disc is the border of one sarcomere. A sarcomere includes the contractile proteins actin and myosin. Myosin is this thick purple, or what we call the dark protein or the thick protein. And you'll notice that it's got little heads on it, like these little bumps all along it. Then, on the edges, this orange protein is called actin. So this is your thin protein, this orange stuff. And then titin is just what keeps the actual actin grabbing on to the Z disc. So this actin right here, what it's going to do when it contracts and you create tension with the myosin, the actin is going to move towards the middle and move towards the middle. So the Z discs actually move closer together. So when I ask you about the contractile proteins, what you need to remember is that myosin is the thick protein and actin is the thin protein that slides over the myosin and then is going to shorten the sarcomere. When you look back at this image, you see how there's sarcomeres. They're all lined up like little boxes all along the cell. And so what happens when these contract is that they will contract rhythmically all together in a pattern, what is going to bring this closer together and shorten that muscle cell. And then what it does is it's having a contraction, which is what's going to generate the force and the pressure that you need for that ventricle or for the atria to move blood from one end to the other. When we zoom in on the actual proteins, so this is your myosin, this is your actin. The myosin is the thick protein that has these little heads. So they have a tail and a little head. This myosin protein, the little head, has to connect here to the orange on the underside and over the top side. But right now, there's no way for them to connect because they've got this bar right here in the way. This bar is a regulatory protein. So is this little yellow stuff. So this yellow stuff is called troponin, and then the bar is called tropomyosin. And these are called the regulatory proteins. And why we care about these is because when you zoom in on this picture, we'll jump back to that one in a second, calcium actually will go and find the troponin and attach. And why that matters is because the troponin is attached to the tropomyosin, which, when the calcium lands on troponin, it triggers the tropomyosin to get out of the way and expose what are called the active sites of the actin, and then the myosin head, click, connects to the actin, and then we have tension. And then in order for the actual shortening to take place, now what has to happen is this myosin head comes off, and then it will click onto the next one and then move the other one along. Then just to go back to here, what I want to show you is obviously here is where that connection is that triggers the whole action potential from the neuron, and then hits the receptors, depolarizes the membrane, then you get your voltage gates for sodium opening up. So you have your depolarization action potential that happens along the way. Here's the T tubule that when the signal is going down and opening those sodium gates, it will trigger the calcium gates inside the sarcoplasmic reticulum to open, and then the calcium goes in to the sarcomere. So then when you look at this image, the calcium goes into the sarcomere and then will attach to the troponin little protein, the regulatory protein, and then the troponin will move the tropomyosin out of the way so that the active sites are exposed and the myosin head can now connect to the actin. So the myosin head, click, connects to the actin. Now we have tension. Now we're going to use ATP. And so now the ATP, we're going to burn one ATP to then move to the next site, which is going to connect, move to the next one. And so then the actin is actually sliding over the myosin because the myosin had clicks and pulls, clicks and pulls, clicks and pulls. And so that is what's going on the entire time when you're having that shortening of the actual muscle. And so those sarcomeres are having this happen, and millions of these sarcomeres all along the cardiac muscle cells, and this is why we can create the pressure. And so then that myosin is going to connect and click, connect and click, connect and click, and that the actin is going to slide over the top. So when you have the contraction taking place, that is called systole. Then once you have repolarization take place, now what happens is the calcium comes off the troponin and it'll go back to that sarcoplasmic reticulum for storage. And what's happening is now we've got potassium gates that are opening in here. That is what now triggered the calcium to come off and then go back to its storage, and then the calcium will get reabsorbed, and then repolarization obviously will happen to the membrane, and then we'll go through our resting membrane potential again with the sodium potassium pumps, and then everything returns back to rest. So relaxation, calcium comes off. That means that the tropomyosin is going to return to cover up the active sites because now the calcium is not on the troponin anymore, so the troponin can't keep the little tropomyosin bars off the active sites. And so then you see how the tropomyosin returns and then covers up the active site, and then the myosin head comes off and then restores its little ATP, and then it's ready to go again for the next contraction. So when you have relaxation happen of the muscle, that is now called diastole. So your cardiac conduction system, remember that this is autonomic. So our sympathetic and our parasympathetic system are what are going to be regulating what is taking place. And so what is called excitability, the excitability is really, really, really important. And that means that you're going to have that depolarization action potential taking place. And remember that the action potential, the depolarization, has to do with the conductivity and the ability of those cells to control the electrical signal. Now, the thing that is kind of crazy about the heart is that we have what is called automaticity. Automaticity means that not only can the heart cells do their thing in terms of being depolarized and action potentials generated by the cardiac conduction system, which is usually the boss, but it can also happen on its own. So cardiac cells can actually generate their own electrical signals, and there can be kind of little brats when that happens, because that is when you start having arrhythmias. And sometimes obviously they can be very, very serious because they start having their own little party in the corner, disrupting the whole thing, and then a person can actually go into cardiac arrest because of an arrhythmia issue, or they can have very, very irregular heart rates. They can either speed up, or they can be very slow, or they speed up and slow down, and speed up and slow down. So it can cause a lot of problems. So intrinsic, what this means is that we have this network of what are called non-contractile autorhythmic cells. So embedded within the actual cardiac cells, we have the conduction system. And the pacemaker, which is called the SA node, is what is going to control the entire sequence of what takes place. The thing about the pacemaker is that it has what's called an unstable resting potential. And what that means is that it never really returns to rest. So setting a resting membrane potential doesn't happen because we continuously have the opening of these sodium channels that happen. So the way the cardiac conduction system is set up is you have your SA node, this is your pacemaker. Then the signal travels to the AV node. From there, the signal travels to what is called the AV bundle, the bundle of His, or the junction, right, where it's going to join into where the space is that the ventricles come in. And then it separates into the left bundle branch and the right bundle branch. And then it goes up into the Purkinje fibers. And so what happens is you'll have your SA node fire, which means it goes off. That sends the signal to the atria to depolarize. And then the signal will continue down to the AV node where it slows down a little bit because you need to give time for that atria, these guys, to depolarize, contract, and push the blood down into the ventricles. So as the signal is slowing down, that contraction and pushing of the blood down into the ventricles to top them off is happening. Then the signal gets down here, and once it hits into this area where you have the AV bundle, it will go really fast down its bundle branches and then get down here to the tip, to the apex, and then boom, it goes up the Purkinje fibers and we depolarize the actual ventricles. So once that happens, now the ventricles will go into systole, they contract, and then they eject the blood on out. And you'll note that as that signal is going down the septum, part of it does have little branches going into these papillary muscles that are going to allow for these chordae tendineae, which are the little cords of the bicuspid and tricuspid valve, to stay tight so that when the ventricles contract, the blood doesn't regurgitate back up into the atria. So, our order: SA node to AV node to the AV bundle or bundle of His or junction, and then down the left and right bundle branches, and then up the Purkinje fibers. So what you need to understand is that the SA node is going to trigger the atrial depolarization. This SA node depolarizes more frequently than any other part of the heart muscle. So that is why it's called the pacemaker. Then the Purkinje fibers are what are going to depolarize the actual ventricles. But we need that signal to come from the SA node all through its AV node, AV bundle, left and right bundle branches, and hit those Purkinje fibers in order for those ventricles to depolarize. So what we have when the SA node is working properly is that it generates what's called a sinus rhythm. So when you hear other medics or doctors say we have sinus, that means that we have a rhythm that is originating at the SA node and is depolarizing the heart the way that it should be. So the pacemaker is working. On average, in adults, this SA node will depolarize between 60 to 100 beats per minute. And as you know, this is normal heart rate for adults. In kids, it's going to be faster. In older adults, it can be a little faster, a little slower, depending on how they're doing. But on average, it's about 75 beats per minute. But the SA node, remember, depolarizes faster than any other part of the heart. The SA node itself, if we were to measure just the electrical potential of those very specific cells, what they have are that unstable resting membrane potential, and that's this little bit right here. This little bit right here never goes fully to rest. It's like when you were in the academy and you're doing your thing, right? And you guys have just finished doing, I don't know, let's say up and down 5 times in the tower. And now you get back down to the bottom and you're like, oh my God, finally we get a little break. And your RTO is like, again. And up you go again. Because you got to go up and keep going, get back down. And he looks at you guys and he goes, again. And now up you go and down. And who knows why they do that, right? Well, in your heart, it's supposed to do this. So what you're seeing here is it gets just that little breath of air, and then boom, it's starting to depolarize. Action potential gets generated. They repolarize, and then they're going back down to rest, and there's like, oh crap, we can't rest again. Now we got to go again. And it just keeps going and going and going. So this is what the SA node electrical activity looks like. This is not an EKG. All right, this is just what happens in those very, very specific cells. And so books always show you this, and they always talk about the SA node having that unstable resting potential because it's really important that it keep the beat, right? It's what's keeping the pace of what's going on. What you see here, this is a normal sinus rhythm. So you have a P wave right here, which is your atrial depolarization. Now the signal slows down a little bit through the AV node, and then as it's going down the bundle branches, boom, it's going to go really fast, and now it hits those Purkinje fibers, and up you get the depolarization of the right and the left ventricle. Then right here between the S and what's called the T, this region is where the ventricles are going to contract. So this is your ventricular systole. And then the ventricles repolarize here with this T wave, and then we have a little flat area. This little flat area is the time when the ventricles are passively filling. So there is no contractile activity whatsoever. So normal sinus rhythm, the definition is there's an upright P wave preceding each QRS complex, and then there'll be a T that follows. P wave, QRS, T. P wave, QRS, T. P wave, QRS, T. And so on. So that's your normal. The AV node, so say that the SA node fails, all right? So now your pacemaker just decided not to come to work. So now something has to do the job. So what takes over is called the AV node, which is the second node in the sequence. However, if the AV node takes over the job, what is going to happen is that the heart rate is going to slow down significantly. The AV node, because there's a signal delay and its job is to continue the signal down to the actual ventricles, it generates what is called a junctional rhythm. And a junctional rhythm is going to be between 40 to 60 beats per minute in an adult. All right, so this is what happens when the SA node doesn't work. You get the junctional rhythm. And just to show you what a junctional rhythm looks like, there's a variety of these, but you'll note that here, where did the P wave go? It's missing. So a real distinct sign of a junctional rhythm is that the P wave is missing. You still have the QRS, you still have the T, but there is no P wave. And so what that is telling you is that the atria did not depolarize. And so you'll have a rhythm that is much slower. Remember that the signal that goes from the junctional rhythm will continue down through the left and right bundle branches, right? But if you get down here to that AV bundle, now things are going to continue to slow down, and eventually we're going to have what's called a ventricular rhythm. Here's another example of what can happen with a junctional rhythm. You can end up with, instead of just a flat line here with no P wave, if the P wave is upside down, that means that the P wave did not follow the pattern, right? So the atria themselves did not follow the pattern of depolarization like they should have. They did it late, all right? Basically went the other way. They started walking away from the direction that they were supposed to be there. It's kind of like, fine, I'm just going to go over here and throw my little fit, right? And so this is your little P wave that's upside down. So an upside down P wave is a junctional rhythm as well. You can also have, in a case where you maybe have like a tachycardia, so it's called a junctional tachycardia, is that you can have a P wave that comes in very late. So here there was no P wave, here's your QRS, and then the P wave came in late and upside down. So here's your T, but basically what happens is you don't come to work, right? There goes the SA node, didn't come to work. Here's your QRS. Then it basically says fine, and it starts storming off the wrong direction, but then comes back and it shows up upside down. So it's kind of like, why do you even come to work, right? So this is your upside down P wave. So you can have a late P wave as well in a junctional rhythm. But clearly you guys can see here that in this slide, right, there is no P wave, junctional rhythm. In this next slide, the P wave is upside down. It's a junctional rhythm. And in this case, the P wave is not only upside down, but it came in very, very late after the depolarization of the ventricles. So these are all kind of the weird looks that you can get when you get a junctional rhythm. So what happens there again, your SA node is supposed to be in charge, but the AV node takes over, and then you can have a whole series of weird looks when that occurs. So now we're going to keep going. We want to get to that bundle and then hit these ventricles. Now if the SA node and the AV node both fail, now the ventricles have to take over. But as you can imagine, now we can have a very slow ventricular rhythm. So when you go into a ventricular rhythm, now you've got some pulses, but they are very, very slow because the ventricles by themselves only depolarize about 20 to 40 beats per minute. So if your SA node, your AV node, and everything in between fails, then the ventricles are going to take over. And so what you end up with is what's called a wide QRS, and there's no P wave anywhere in here. All right? So the ventricles had to take over. So that's what you're seeing here. And this is what's called a wide QRS, which means it took a lot longer for that signal and for the depolarization to happen in the actual ventricles. So that's what occurs here. So that's when this happens, when our ventricles take over. Now, when you look at the actual cardiac muscle, all right? And every time I have students look at these two images, they're like, what does this mean? All right. So what I'm going to show you and what I want to make sure that you understand is remember that we have, okay, an electrical rhythm that is the cardiac conduction system. These are very specialized non-contractile cells that function from your parasympathetic and sympathetic system, all right? The job is of those two branches to control heart rate based on what state your body is in. When your body has to do something, right, and react very quickly, it goes into a sympathetic state. When you want to chill out again, it goes back to being parasympathetic. So we have the cardiac conduction system, where we have the SA node, AV bundle, and the bundle branches and up the Purkinje fibers. And then we have the heart muscle, which is its own little set of tissue. So what you see here, this is that electrical potential of what I showed you earlier that the SA node goes through, which is different, right? These are the phases of that SA node depolarization, and when it generates its action potential and allows for that depolarization of the atria to happen. What you have over here is just the muscle itself. Now, one thing I hate about these two images is that the color that they use to show the SA node pattern and the phases that it goes through is the same color that they use to show the tension in a heart muscle cell. That's what this purple line is. The blue line that you see here, this is the actual signal that happens in cardiac muscle tissue. So the atria and the ventricles. And so what I'm going to go through right now is I'm going to explain what happens in each of these 0 to 4 phases. So it goes from 0 to 4, and explain what is happening in cardiac muscle tissue, so that you understand why this looks so weird, all right? And so the blue is the actual cardiac muscle. All right, so you understand that we have resting membrane potentials. So the heart muscle is at rest. Outside, we have in the extracellular fluid, sodium is higher, potassium is lower, right? And then inside the cell, inside the membrane, we have more potassium and higher concentrations. Sodium is going to be low. Then the sodium-potassium pump, remember, is what maintains the electrical gradient to keep things balanced. So in this phase 0, all right, what is going to occur is that right here, okay, here we have our resting membrane potential. Then when the cardiac muscle cell gets the signal, go, all right, well, let's talk about the atria, all right? So here's your atrial cells. Boom, they depolarize. So now what we need to happen, right? Phase 0 is right here. This is the depolarization of the cardiac muscle cells in the atria. If this was in the ventricles, exactly the same. This is the depolarization of the cardiac muscle cell in the ventricles. So what you're seeing is, okay, there goes the depolarization. So what happens is, just like before, sodium is going to rapidly enter the membrane. You have your depolarization occur, and you're going to start having that action potential that is going to cause the calcium to start leaking out of that sarcoplasmic reticulum and start the contraction phase. So the contraction phase of cardiac muscle is going to follow the depolarization because it happens during the action potential. All right, so now we go to the next one. So now we have phase 1. That's up here at the very top, the first little dip right here. So what happens is we had our depolarization in phase 0, sodium gates opened, and that triggered the calcium channels to start opening. Now, in order to slow things down so that we have the contraction of that muscle cell, right, we need the tension, right, what you see here, this is the muscle tension in the cells building up, right, so it starts to go up because this is allowing for those little calcium channels to start to open. When you get here to phase 0, how we're going to slow things down, or we're going to start what's called an early or initial repolarization. So that's phase 1. So what happens here at the very top, we have some closing of sodium channels, and simultaneously what is happening is we have slow potassium channels opening, and we have chloride channels opening. So right here where the little arrow is in phase 1, we have potassium is leaking out of that cardiac cell, and we have chloride channels that are open, and look at chloride is negative, chloride is actually entering into the cell and starting to get the inside of the cell to become more negative as potassium is leaving the cell, which, as you know, also allows for a cell to become more negative on the inside, more positive on the outside. So this is like you're starting to slowly apply the brakes a little bit, because now what's going to happen next is this plateau phase here. And why we carry the plateau phase, phase 2, this is when we get more calcium channels opening and more calcium is going in to the from the sarcoplasmic reticulum and leaking out into that sarcomere and attaching to the troponin, so that that tension, boom, can build up in the cardiac muscle cell. If we didn't have this, where the calcium channels are continuing to open and that cardiac muscle cell has that calcium attaching to the troponin and moving the tropomyosin out of the way so the head of the myosin can click onto the actin and start doing its thing, we need this right here to happen in phase 2. So this is that plateau phase, right? And this is also where you have that peak cardiac contraction of the muscle. Once this peak cardiac contraction has happened, now obviously the muscle has to relax so we can do it again. So now what happens is phase 3. Boom. This is normal repolarization that you know, where we have all the sodium gates, the calcium gates closed. So calcium is going to return back to its storage unit. Sodium gates have closed, and now the potassium channels open. Boom. And out rushes potassium and repolarizes the cardiac muscle cell again, right here. Then we're going to have the relaxation of our muscle. So if this is a ventricle, now that muscle is going to be entering into diastole. If this is the cardiac atrial muscle, then the atria would be entering diastole. Then we go back to phase 0. This is where the cardiac muscle is now completely relaxed, whether it's in the atria or in the ventricle, and now we could start again to passively fill. So right here, this is where we now restore that resting membrane potential. The muscle is relaxed. We're in diastole now, and now the blood can start returning into those chambers. So that is what takes place in the 4 phases of the cardiac muscle itself. So when we look at it collectively, again, phase 0, that's this one here, we have the depolarization, sodium influx via fast voltage gated channels. Boom, depolarization. We're generating our action potential, which is very important, because what is going to happen here is obviously we need a little bit of slowdown. So here we have a little bit of potassium starting to leave the cell, chloride channels open, a little bit of chloride is starting to come in during phase 1. Then we go into the plateau phase, where we have calcium channels now opening, and the calcium is going to the troponin, landing on the troponin, allowing for the tropomyosin bars to get out of the way. The myosin head attaches to the actin, creates tension, and now we contract that muscle fully. So here's the full contraction of the muscle taking place. Once that's done, we've contracted our muscle, we ejected the blood, boom. Now we're going to repolarize, so that's the phase 3. And in phase 3, we have the sodium channels closed, the calcium goes back to storage in the sarcoplasmic reticulum, the calcium channels close, and we have those potassium channels opening. Boom, so here comes repolarization. Then we get down to phase 4, and at phase 4, now we have the restoring of the resting membrane potential. So up here, where we had the cardiac muscle contraction tension at its max, this was your cardiac systole. And then down here, where we now restore that resting membrane potential, now we have diastole. And then what you'll note on here where it says absolute refractory period, what that means is when something is refractory, you can't make it do something at that moment. Or in some cases with medications, it means the medication did not work. So what you're seeing here is when you have depolarization action potential and that contraction happening, when the cell is already in that state, you can't make a depolarized and contracted cell do it right then and there because it's already doing it. All right, so you can't stack them together. So this absolute refractory period essentially means absolutely not, under no circumstances, can you contract a contracted muscle cell. All right, And so this also is exemplary of what happens with a toilet. So for example, you flush the toilet, the water goes out. And now you can't flush the toilet on top of the flushed toilet because it's already flushing. So that's why this absolute refractory period, you can't contract a contracted muscle. All right? It's just not possible, no matter what you do. But once you get down here at the end of what's called the absolute refractory period, now when we're establishing that resting membrane potential, this is where we have what's called relative refractory, and that is where that membrane is hyperpolarized, and we could, if we needed to, or if the heart feels like it, because it does sometimes, it will jump ahead and it'll throw in a beat, sometimes out of order, or if your heart is going very fast, it'll just jump in. So during relative refractory, you can, during this phase 4, actually stimulate that muscle cell to contract again. All right? So these are the phases of the cardiac muscle contraction. That is what an absolute refractory period means and what a relative refractory period means. So again, when we go back to look at this, you can now see all phases together, and I've got them summarized here on the left. And then you can see that what happens in the SA node and its little depolarization and sequence of electrical potentials is very different from what you're seeing here in the blue that is actually taking place in the actual cardiac muscle. All right? The SA node itself actually doesn't really have a phase 1. All right? It goes zero depolarization, boom, right? And then very quickly, very minimal here. We don't really have a phase 2, and then it repolarizes. And then again, it doesn't have a real true resting membrane potential. When we look at the individual nodes, so if you wanted to look at the individual SA node, right, this is what we just saw back here. So this is what you're looking at here in terms of what does its electro voltage looks like. Then the atria look more like cardiac conduction, right? Boom, depolarization. Here we have early repolarization happening. This is when your action potential and your contraction are going to start to occur. And then boom, repolarization, then back to rest. And then we have the AV node. So the AV node has its electrical activity. So then it depolarizes. But you can see that the actual way that the AV node does its thing, it takes a little bit longer, right? This space here is longer, took more time than what happened up here. This was very quick. And so what you're seeing here is that slowdown in order for the atria to be able to contract. Then once the AV node does its thing, right, the signal is not going down into that AV bundle. So the AV bundle is going to receive its signal. It's going to carry that signal through the left and right bundle branches, which is going to be here. And so they're going through, and then the Purkinje fibers receive the signal, and then they're going to depolarize the ventricles. So you can see that the ventricles themselves have a very similar pattern to what the Purkinje fibers are doing. But it's here where we have that depolarization of that cardiac muscle, and then we have that little slowdown, the contraction phase, the peak, right, contraction, and then repolarization of the ventricles, and then we return back to rest. And so what you're seeing here is that composite of what is taking place with the SA node, the atria, the AV node, the AV bundle, the bundle branches, the Purkinje fibers, and the ventricle. So here, this is as our SA node is starting to fire, boom, we depolarize the ventricles. You have the atria doing their thing. Atria depolarize, then they repolarize. And as they're repolarizing, right, we had the AV node pick up the signal. Things are going to slow down. Cardiac or atrial systole happened here. Now they send the signal down into the bundle, boom, down the bundle branches, so very, very quick. And then it hits the Purkinje fibers, and then boom, the ventricles depolarize. And then what happens is once those ventricles depolarize, now we're going to have our contraction, which is right here. So this is your ST segment, and we have the contraction of the cardiac tissue, and then the ventricles with the T right here. Now these guys are going to repolarize. So essentially, that's what you're seeing right here. So that's your cardiac conduction system and what is taking place in the actual muscle. Now what we have here and what you're looking at is basically the medulla oblongata here. Remember that this is where the regulatory system is that's going to regulate the sympathetic and parasympathetic branch that is going to innervate the heart. So in the purple, this is your vagus nerve. So this is your preganglionic neuron going down, and The parasympathetic system with the vagus nerve only innervates the SA node and the AV node. So anything that is going to affect the SA node and the AV node is what's called supra means above, ventricular, which is above the ventricles. So you'll note that the brakes, which are the actual parasympathetic system, are going to apply the brakes gradually to the SA node and the AV node so that the signal gradually slows down. And the reason why this is like this is because you don't want to, like, bam, slam on the brakes, right? That would not be good. You'd pass out. So, but when your body says, boom, we need to go into a sympathetic state, we need to go now. Now we have this little interneuron that comes from the cardioacceleratory center, which is your sympathetic, and it hits that preganglionic neuron that originates from the lateral horn of the spinal cord, goes out the ventral root, because remember this is motor, and now it's going to go up to its little ganglion, synapse with the postganglionic neuron of the sympathetic system, and now you see that we've got connections, sympathetic postganglionic connections from that neuron going to the SA node, the AV node, and the ventricles. So this is why we can very, very quickly speed up our ventricular function in our heart, but you don't want to slam on the brakes. So where you see cardioinhibitory, this is the vagus nerve, slow things down. Cardioacceleratory, this is your sympathetic branch, speed things up. So on the EKG, what you have is telling you what is going on in the heart. All right, and I kind of previewed that a little bit earlier when I showed you what happens with the leads and what happens with the actual arteries. So EKG paper is designed the way it is because it's giving you information, but it's information based on conditions. Conditions based on signal amplitude, so how strong or how tall the signal gets, and how long did it take for this signal to happen. So that is why you have these. So each little box here is 1 millimeter by 1 millimeter, and the length of one little box here is 0.04 seconds, or what's called 40 milliseconds. Its altitude is only 0.1 little millivolts, right? So 1 millimeter is 0.1 millivolts. Then you'll notice that we have one big box. That's what we call it. So we have these darker lines. So one big box is 0.20 seconds, or 200 milliseconds. And the actual time in terms of what's passing through and all that is 0.5 millivolts of the actual amplitude. So 0.5 millivolts is this. 1 millivolt would obviously be 2 of these. And so the amplitude is going up, the time is going across to the right. And why this matters is because the length and height of the P waves, the QRS, and the T, and how long everything took is going to be indicative of some things that are going on with the person's conduction system based on the pattern that you see on the actual EKG. So what you want to remember, and you need to memorize this, is that the P wave means atrial depolarization. So what that tells you is the SA node fired and sent the signal to the atria, and the atria depolarized, and boom, they went into their systole and contracted, pushed the blood down through those valves into the ventricles, topped them off, right? We want that. We want atria to depolarize because we need the atrial kick to top off the ventricles with the blood that is in the atria. Then we have this PR interval, and we have a PR segment. So I'm going to get to that one in a second. And then the QRS complex, this is your ventricular depolarization. So that means the signal got from the SA node to the AV node, down the AV bundle, through the bundle branches, and bam, it hit those Purkinje fibers. So that's your QRS. Following that is the ST segment, which tells you ventricular systole, and then the T wave tells you the ventricles repolarized. So we want this all to be exactly right, very pretty, just like you see here, because this is going to be a good EKG. This is what you see, normal sinus rhythm. So to zoom in a little more clearly, here we have our P wave. So here is our atrial depolarization. So SA node fired here, boom, atria depolarized. Now we have that contraction, the atrial kick between the little PR segment, and now that's going to follow, goes down, right, hitting that AV bundle, down the bundle branches, boom, hits the Purkinje fibers. So that is our ventricular depolarization. So that's your Q here, R, S, and then the ST segment goes from here to here. All right, so from where the S is, boom, over to the T, and then this is where the ventricles are going to contract. And so what you don't want to happen is this little piece to be elevated, because then that would mean you have an ST segment elevation. All right, and then over here with the T wave, the T is showing you that now your ventricles repolarized. And so what you're looking at here in terms of how long should things be, right? These are your normal, basically, benchmarks of what you want, what is normal for a PR interval. And a PR interval, this is important because it's telling you the atrial depolarization, the atrial systole, and how long the signal took to get to those Purkinje fibers. All right, that is why we care about this. If this is too long, it's going to take longer for the signal to get to the QRS. Or in some cases, sometimes the signal just never gets there, right? It just skips, and then the ventricles don't depolarize, and then your patient's going to be like, oh, that felt kind of weird. All right, then what we want here is that ST segment, because remember, what we care about is that the ventricles contract and send the blood out, because that is where we're getting our pulses. And so very, very important for that to happen. So this is where we look for those STEMIs, right? The ST elevation. And then the T wave is going to tell you, are the ventricles repolarizing? But what the T wave also tells you is what's going on with our potassium levels. So sometimes you can have inverted T waves, which means low potassium, or you can have what's called peaked T waves, which means high potassium. So you'd be hyperkalemic. So normals for the PR interval are going to be from 0.12, so that means 3 little boxes, to 5 boxes. So that's 0.12 to 0.2. If it's longer than 0.2, that's a type of heart block that you're having. Then the QRS, normal here in this textbook, they call it 0.08 to 0.10. I've seen 0.08 to 0.12, 0.09 to 0.11. It just depends on the book you're looking at. And then you've got your ST segment, right? Because we want this to be not elevated. And then the QT interval is very important. So here's the Q, and then we get to the T. And why this matters is this is also going to be indicative. How long did it take for those ventricles to depolarize, contract, and then come back and repolarize? So that's really important. And then from R to R, this is counting heartbeats. And so this normal interval is going to be 0.6 to 1.0. And this would be a normal heart rate. So P wave, atrial depolarization, and it was initiated by the SA node. Then you're going to have the signal passing through the AV node. So your atria depolarized, you have your atrial systole happening, and the signal is slowing down through that AV node to top off those ventricles. Then we get the signal passing down, AV bundle, down the bundle branches, and now we're going to hit ventricular depolarization, which is your QRS. Following that, we're going to have our ventricular depolarization is done. Now we have our ventricular systole, which is the contraction. Boom. And now we're going to have the T wave, which is going to be ventricular repolarization. And now we're complete. And now we're going to have our little flat line in between here. And that means passive filling. And so we're ready to go again. So here it is all together. So remember, P wave means atrial depolarization. Then we're going to have the little segment where we have the atrial systole. Then we have our QRS, which is our ventricular depolarization. And then from there we have our ventricular systole. Then we have the T wave, which is ventricular repolarization. And then everything is back in diastole. So the cardiac cycle itself, right? We have the electrical events. Now we're talking about what happened from the SA node to the AV node, down the bundle branches, and then up the Purkinje fibers. So what you're looking at here very specifically is showing you exactly what takes place. P wave, atrial depolarization. Following the P wave, atrial systole. From atrial systole, the signal, the little tiny deflections called the Q wave, that signal is now sending down from the AV bundle, boom, down the bundle branches. then the R wave, now the ventricles, right, from Q to R, boom, ventricular depolarization is starting, and we get down to the S, those ventricles depolarize. Now we're going to be between the S and the T, so now the ventricles contract, so that's your ventricular systole, and then you're going to have your ventricular repolarization with the T wave, and now we go back and ready to go again and rest. And so you may be asking yourself, okay, well, if the atria depolarized and they went into atrial systole, when do they go back and relax? And so when they go back and relax and they actually repolarize during the QRS complex. So when the ventricles are depolarizing, that is when the atria right here are entering their repolarization. And you see right here, they're repolarized, and now they are going into atrial diastole. So the actual atria are actually going to relax before the ventricles go through their process. So they start entering diastole as the ventricles are depolarizing and going into systole. So when we go back and we look at our 12 lead and the EKG and the coronary arteries, now you can look at this and be like, wait a second, okay, we have a P wave, we have our QRS, but now we have a little kind of inverted little area here that is showing some ischemia somewhere, and now here's our T wave. But now we look over here to where the actual MI is going on, the myocardial infarction, and you can see very clearly, here's our P wave, boom, boom, boom. The Q looks really weird, right? And the R, okay, and here's our ST. So everything is messed up right here, specifically right here. It looks really bad there. So this right here is a classic ST elevation, or what we call a STEMI. So back again, let's look at where that happened. V1, V2, V3, V4. There is an issue going on in the interventricular septum and the anterior wall of that left ventricle. So what this is telling you, interventricular septum, this is where that AV bundle and the bundle branches are located, and where that beginning of ththose Purkinje fibers are. So this is not good, all right, because this piece of the heart muscle is dying. And so it is really important that you catch these things, that you get really good at learning these EKGs. And when you go through cardiac and you're going through the EKG stuff, you will see and learn very quickly how to pick out these STEMIs. There's probably the easiest ones to actually pick up. So back here, right, here you see a little bit of weird kind of ischemia going on, which means there's some decreased blood flow somewhere in it. And yeah, I mean, it would actually make sense for that to be happening, because what you're seeing here in 1 AVL, right, so right here, this is where the LAD is. This is where the biggest problem is coming from that LAD. And then remember that you're going to have that LAD that is going to hook up and it's going to unite back here. And we've got blood that comes this way and then gives into this bottom portion and inferior portion of that ventricle. But the big issue is right here in V1, V2, V3, V4. All right, that's where you see that ST elevation that is just weird. And so that's where it's telling you that this is where that part of the cardiac muscle is being impacted. And the other big problem with that is this is left ventricle. And remember, it's your left ventricle that shoots the blood up into the aorta and leads over to that systemic circulation. So we can't have that die. All right, and so that is why it is so important to be able to get this person as fast as we can, safely, to the cath lab, because that is where, hopefully, if they're alive, we'll be able to get the actual blockage cleaned up and the person will survive. But it doesn't always happen. All right, so in terms of heart sounds, so what are the valves doing? So we've talked a lot about the valves and where they're located and what they do. And so when you listen to the heart, you hear this little lub-dub pattern. The first one that you hear, the first heart sound, which is called S1, is the closing of the bicuspid and the tricuspid valves. So remember, the bicuspid is also known as the mitral. And this is at that start of when the ventricles have depolarized and now they're going to contract. The S2 sound, the dub, this is the closing of the aortic and the pulmonary valves at the end of the ventricular contraction. So if you happen to hear a little whoosh, okay, you hear S1 closes, but then a shoo during S2, that would be telling you, hey, something's kind of weird going on here, because the first heart sound, That's the closing of those bicuspid, tricuspid. And then S2 is the closing of the aortic and the pulmonary. But if there's a sound somewhere in between here, because now when the ventricle is contracted and out went the blood, but now you hear a little whoosh, something is going on where now maybe one of these cuspid valves is not closing all the way. All right, so when you look at the heart and the chest itself, to listen to the specific valves, and again, this is something you as a medic are going to do in the field, listen to all 4 areas. Most of the time when you listen to the heart, you're putting the stethoscope here just on the upper portion of that left side of the chest. And what you're actually kind of listening to, and you can hear really well right here, is the pulmonary valve, and obviously you also hear the AV valves. But when cardiologists are actually doing a cardiac test and listening to all the valves, they will go through and they will place the stethoscope on all 4 areas to listen to what's going on. So on the upper portion, this is where you're listening to the aortic valve on the right, because that's the direction that the aortic valve is when the left ventricle pumps against it and shoots the blood up into the aorta. Up on the left, that is the top left, that is going to be the pulmonary valve. So the pulmonary valve is up on the left, and then if you want to listen to the mitral, it'll be on the bottom towards the left, which is going to be the bicuspid, and then the one on the right, your tricuspid, would be on the bottom right. So that's where you would listen to the particular heart sounds in terms of where they're located. All right, so the next one, S3, these are abnormal heart sounds, so you shouldn't really hear any abnormal heart sounds. So if you hear this low-pitched, third way through what's called diastole, like when the actual ventricles are relaxing, it would be like lub dub da, all right? And then sometimes you'll hear this S4, which is right before S1, which sounds like a blah lub dub, okay? And you can go on YouTube and you can hear these different heart sounds, but what they're telling you is there's something going on. There's just weird murmurs, all right? So the most common murmur is going to be usually between with the mitral valve, all right? Sometimes there'll be a louder murmur that occurs when there's an abnormality with the mitral valve, the bicuspid, and the aortic together. Bruits, these are specifically sounds that you hear in the carotid arteries, and that is not something that we as medics are going to be evaluating for people, but you'll probably notice that they do listen to your carotids when you go to the doc to get your physical done. And then any clicking is usually going to be those high-pitched sounds when the valves open maximally. So when those ventricles contract and are pushing the blood out, that's when you would hear any kind of weird noises if there was dilated aorta or pulmonary trunk, which means that there's some bubbling maybe that's taking place in there, or what's happening there is also sometimes the bicuspid is really, really flexible, so it's kind of floppy. And then the snap, that can sometimes occur when the valves are stiff, and now you're having to really push through and like kind of snap it open, right, when you just kind of flick it with the pressure of the blood coming down from the atrium. But most common, the 2 sounds that you have to remember what they are murmur, and also make sure that you know the bruit. When you guys are doing the cardiac emergency chapter in the medic stuff, you will for sure probably get questions on these in those JB learning tests. All right, so here's our cardiac cycle sounds. So remember systole to diastole and repeat. So that's where we're going to have our normals. So S1, lub, means the closing of the bicuspid and tricuspid valve. S2 is going to be the closing of the semilunar valves, the aortic and the pulmonary. And then your abnormal sounds, primarily you will hear them more during these S3, S4, which is going to be happening during that phase of diastole. All right, so the cardiac cycle. The cardiac cycle is what we've been talking about pretty much, which is the onset of the muscle contraction, and then we get the muscle contraction, we go through the full phases, we relax, we fill, and then we do it again. And so how a muscle works, right? It contracts, it creates pressure, and volume should go down. So this is where it's really important that you understand the relationship between pressure and volume. So if pressure goes up, that means volume is going down. So if you think about the heart, when the ventricles fill, there is low pressure because the actual ventricle is in diastole. It is not contracting. But the volume of the blood in the ventricle is going up. So that's what I'm talking about volume goes up when pressure is down. So during cardiac diastole, volume of blood is increasing in the chambers. During contractility, which is systole, pressure goes up and volume goes down because you're ejecting blood. You're moving it from one place to the other. And so the pressure gradient that is created from the actual systole of the muscle cells is what causes your gradient to go from high pressure to where it's in low pressure. So the blood moves from where it was located in the ventricles, right? And we squished it, so we created high pressure on that blood, and it moved from where it was in a high volume to where it's in the lower volume. Or we exerted high pressure with the contraction, and it moved to where the vessels were relaxed, which is where the blood is going to flow into. So atrial systole, remember, is the contraction of the atria. Their job is to top off the blood in the ventricles. And at the end of that topping off, that you have the ESV, which is the end diastolic volume. Then the atria will go into diastole. The atria are going to relax, and this is happening when the ventricles are depolarizing. So you will never see on the EKG the repolarization of the atria. It just isn't something, it's not a signal that we look for. The ventricular systole, this is the contraction of the ventricles. So now they are pumping the blood into the systemic circulation and obviously into the pulmonary. So now they are increasing pressure on that blood, and now that blood is going to be ejected out of the ventricles and move into the vessels. So what is called isovolumetric contraction. It's an important concept that you understand. This is that split second right before the ventricles contract. The ventricles are filled to the max. All 4 valves are closed. It's a very important thing you got to remember, right? So all 4 valves are closed. We've topped off the ventricles, and now we are about to contract. So the way I always remembered this, think of it as iso ready to contract. So isovolumetric contraction means the ventricles are about to contract, they are full, and all 4 valves are closed. Now the ventricles contract, they eject the blood out, and that blood being ejected from the heart. So what we measure on that arm is going to be systolic blood pressure, which is systole, right? So systole, boom, ventricles contracted, blood exits the heart, and it goes into that aorta. And as you know, we also have blood from the right that goes into the pulmonary trunk. Then the ventricles are going to repolarize. So when the ventricles repolarize, they go into diastole, but right before they relax, there's that one little split second that's called isovolumetric relaxation. So again, think of this as iso ready to relax. So this is that little phase right before the ventricles are going to relax, where again, all 4 valves are closed. Because remember, what just happened was those ventricles contracted, ejected the blood out, the valves in the semilunars, right, your aortic and your pulmonary valve are closed, and right as that heart was contracting, we also had the bicuspid and tricuspid valves closed. So that's why these 4 valves are also closed during isovolumetric relaxation. Then we relax, which means we're going to go into diastole, and now what that does is it generates your diastolic blood pressure, which is the resistance or pressure of the blood flowing through the systemic arteries as the heart is relaxing. So your diastolic blood pressure is that bottom number when you're doing a blood pressure, right? And that number should always be lower than the systolic. So here's our phases. Again, this is not what a normal EKG would look like, because obviously these are really far apart in terms of the way the waves are, but it just kind of shows you what is happening in terms of the conduction system and then systole and diastole in the chambers and their muscle. So the P wave, remember this is atrial depolarization. It's going to be followed by atrial systole. So between your end of your P and beginning of your Q, right? This is when the atria contract. Then you're going to have QRS goes off. This is your ventricular depolarization. When this happens, that is when your atria are now entering into that atrial repolarization, and then they're going to go into diastole. When the ventricles get depolarized, their next step is they're going to contract. So now they go into their ventricular systole. They contract, that's your S to T. Then the ventricles will repolarize, so that's your T, and then they will enter into diastole. So then you have, at the end of that T wave, the ventricles are entering diastole, the atria are already in diastole, and what's also been happening is that blood has been returning, right, because your atria went into diastole here. So now you've got your atria here that have been filling, right, blood's been returning, and they're just waiting for those valves, the tricuspid and the bicuspid, to open when the ventricles go into diastole. Once they go into diastole, these valves are going to open, the blood is going to flow passively into the ventricles, and that is what you see right here, between the end of the T and the beginning of the P. And then boom, we do it again. What blood remains in the atria, they contract, top off the ventricles, so that's your P, atrial depolarization. We have our atrial systole, right? And then we go back again and repeat. So that is what's happening in terms of the filling and all of that goes on with the heart. Then when we look at our phases, right, we have our ventricular filling phases, we have our ventricular systole, we have our early diastole, and you'll see here we have, the phases of isovolumetric contraction and ejection and all of that. So when we get into the big picture, right, if you take a line or a ruler or something and you kind of draw it straight down, you are able to see simultaneously what is happening. What is the EKG doing? What is happening with the valves? What is happening with pressure, right? So the systole of the atria and the systole of the actual ventricles, and when the atria enter into diastole and when the ventricles enter into diastole. So this section of the chart is pressure, and then the bottom section is volume, which means blood volume. So here you can see the atria are filling, right? This is right before the P. So this is your passive filling phase. The atria are relaxed, they're in diastole. The ventricles are also in diastole. So passive filling. Then we have P wave, boom, depolarize the atria. The atria go into systole, and they top off the ventricles. So your EDV goes up, right? End diastolic volume, this is going to increase. Then, obviously the valves, tricuspid and bicuspid, are going to close, because now we have the QRS, which means ventricular depolarization. Now the ventricles are going to go into systole, and the atria are going into diastole. And now what happens, because you had your ventricular systole, now the volume is going to go down because the blood is being ejected out of the heart. And what you also see here in the green, this is the pressure of the blood in the aorta. So as that blood is ejected out and it goes into the aorta, you can see that the pressure in the aorta obviously increases. Now, once the ventricles have done their thing, they're going to go into diastole, and obviously the volume had been emptied. So when the volume emptied at the end of the contraction, sorry, I forgot to mention this, at the end of systole, that is when we end up with ESV, which is our bottom number, the volume of blood left in the heart at the end of, after they contracted. So here we go into diastole. Here is our repolarization of the ventricles. Our semilunars have closed. We're in diastole, and now we're able to start filling again. And then here is where you see the blood leaving the aorta and being distributed. Then those phases that I talked about the isovolumetric, so that is actually taking place right here. So this is right before, remember, isovolumetric contraction means iso ready to contract. So it's right before they contract, all four valves are closed. It's right after we had our atrial systole here, and right before you're going to see the actual atrial systole portion with that ST segment. Then the blood got ejected right out, and then right before we go into the diastole here, now what is happening, you kind of measure that up, and this is the isovolumetric relaxation phase, which means the semilunar valves are closed, the cuspid valves are closed, and we are iso ready to relax. Then these, the bicuspid and the tricuspid valve relax, they open up, the ventricles are in diastole, and now the blood that was returning to the atria are going to passively fill into the ventricles. So that is your cardiac cycle. This next part of what we're going to be getting into talks about what is happening in terms of blood flow, and then this very important concept of what's called perfusion, which obviously has to do with, did blood get to the organ, right? And the only way the blood gets there is going to be through the arterial network. So the arteries have to be intact, and obviously once you get down into the capillary, you want to make sure that the actual gas exchanges can happen, which is very important. So cardiac output is equal to the stroke volume times the heart rate. And so this pretty much means how much blood is going to be circulating throughout the body and then returning back to the heart per minute. So stroke volume is the milliliters of blood that are pumped every time the heart beats. And then the ejection fraction is the percentage of the blood that's ejected from the ventricle during systole. So what that means is not 100% of the blood that gets ejected is going to go out of each of the ventricles. So as we talked about before, we had the end diastolic volume, and then we had the end systolic volume. So the ejection fraction is basically the end systolic volume divided by the end diastolic volume, and that tells you how much went out. So your heart rate is measured in beats per minute. So every time the heart beats, there's a volume of blood that gets ejected. Then the cardiac reserve is going to be the difference between your resting and your maximal cardiac output. And so that basically is what we have in between, right? At rest, remember that it's the parasympathetic system that is taking care of things. So this is your brakes. So your cardiac output, when we measure it in liters per minute, is going to be the heart rate, obviously, times the stroke volume that happened per beat. So on average, it's going to be about 525 liters per minute, or 5,250 milliliters per minute. And on average, people's heart rate goes 75 beats per minute, and then the milliliters per minute or per beat that get ejected out of that ventricle are going to be on average about 70. When you're working out, you can actually reach up to about 35 liters per minute passing through the heart because of the increased volume that is always returning, and then of course the heart rate is going so much faster. So that is why during a sympathetic state, the cardiac output goes up significantly. So key factors in terms of the cardiac cycle are this first thing that's called preload. Preload has to do with the degree of stretch in the cardiac muscle prior to contraction. And this is going to be a function of the venous return from the pulmonary and systemic circuit. So when that blood returns through the veins and enters into the ventricles, once that atria have contracted and then the blood in the ventricle is topped off and you've got your end diastolic volume, that's going to stretch the heart a little bit, just like a water balloon that is being filled with fluid, right, with the water. So that is called the Frank-Starling law of the heart. And so preload relates directly to that degree of stretch in the cardiac muscle just prior to contraction when the heart is full. Then the contractility is the actual force of contraction. So how much force did those cardiac cells generate when they contracted to eject the blood out? If the heart becomes weak, or if something happens where the cells are too stretched out, then that would actually cause a myopathy, which means something is wrong with the heart. Afterload is the pressure or resistance in the aorta or the pulmonary trunk or the arteries and the systemic arteries that must be overcome for the ventricles to be able to eject the blood. So basically it is, what does the ventricle have to contract and push the blood through for the blood to be able to get out? It's the stuff that exerts resistance. So if you had two people and they were pushing each other and trying to get over a line, each person is going to exert a certain amount of resistance. But the person that exerts the least resistance is going to lose. So the ventricle, if you think about it, the ventricle is the stronger person, and then the resistance that the person that is a little bit weaker is exerting is the resistance the stronger person has to overcome. So it's that force that the ventricles have to overcome against what's on the other side. And so the other side of the blood, so it could be the valve, it could be the vessels, or it could be, for example, fluid in the lungs, right? That would increase afterload. So when afterload goes up, it makes it much harder for the blood to be ejected out of the heart, and that's when the heart has to contract more forcefully. And the only way to increase that force of contraction, obviously, is going to be that the heart has to get thicker. And so that can cause problems in terms of, if it's on the right side, then that could cause hypertrophy of the right ventricle, which is not good because then it can actually decrease the amount of space that the volume of blood has. And then if the left ventricle becomes pathologized, and then there's an issue there in terms of the thickness of the heart muscle on the left ventricle, then that also can cause problems. So abnormal pathological hypertrophies are not a good thing because eventually it's going to cause problems as a whole for the individual that has that issue. So your preload, in looking at this image, is the blood that's going to be returning to the heart, right? And then it's going to stretch it. So your ventricles are going to stretch. Then the afterload is, what does the ventricle have to overcome in order to push the blood through? And then what you see up here, this would be an example of what would be happening if there was some kind of occlusion or narrowing, or if the vessel was becoming thicker, right? It's going to be harder. It's going to increase pressure on the other side of that blood in order for it to push the blood out. So preload, again, dependent upon venous return. If venous return is increased, then that means more blood comes through, preload increases, there's more stretch, and it's going to have a more forceful contraction. When you have decreased venous return, then the heart is going to contract less forcefully because there's less blood and it doesn't stretch it as much. So anything that interferes with the venous return, the blood returning back to the heart, either on the right or on the left or on both, is going to directly impact preload. So shock will always cause a decrease in preload. Contractility, again, is a function of the calcium levels and the hormones. So the hormones we're talking about, of course, are going to be your catecholamines, epinephrine, norepinephrine, and dopamine. And then you've got your thyroid hormone, which is also very important in terms of maintaining that contractility in the heart. Then the afterload, again, is having to do with the resistance that's in the vessels, or if there's any fluid in the lungs, or if something's going on in terms of what's going on in the arteries. So the afterload is what can then cause hypertrophy in those ventricular myocytes. The atria can also hypertrophy if there end up being issues in the actual bicuspid and tricuspid valves, or if there's a problem in terms of the way the ventricle is set up and now there's too much volume inside the ventricles, then the atria would have to contract more forcefully to try and push blood through. And then pathology can result when obviously you decrease the amount of actual space in the ventricles because they hypertrophy too much, and then it makes it too difficult to get the right volume of blood in there, and then it ends up with issues down the road. The medulla oblongata, again, is where your cardiac center is located, along with the vasomotor that controls your blood pressure and your respiratory centers. So the cardiovascular center, remember, is your sympathetic gas, and it innervates directly the SA node, the AV node, and then the myocytes of your actual ventricles and the atria, and then also the coronary arteries, which are very important, obviously, to actually get the blood supply that you need for the heart. The cardioinhibitory center, that's your vagus nerve. It's your parasympathetic. It's the brakes. And this is what actually will go and directly connect to the SA node and the AV node, and then that will cause an inhibition of the SA node and AV node, thereby hyperpolarizing some of those cells and causing those cells to depolarize less frequently. So if a cell depolarizes less frequently, then it can't contract as frequently, so that slows things down. So that's your brakes. So when you look at it again, this is your medulla oblongata. Here's the vagus nerve, so this is going to be the brakes, and those are connected directly to the SA node and to the AV node. There is no parasympathetic innervation that goes to the actual ventricles. Then to speed things up, that is going to be your cardioacceleratory center, and then that is all sympathetic. So when you look at the sympathetic postganglionic neuron, it goes directly to the SA node, AV node, and also to the ventricles. So this is where you're going to have that really fast speed up, bam, you can just hit the gas and off goes the heart, it starts beating faster. But your vagus nerve is what's going to gradually decrease in the heart, and then gradually put on those brakes and gradually slow things down. The chemoreceptors, again, that we have for the cardiac innervation are going to be, you have your betas, right? So you have beta 1 in the heart, those are adrenergic. In the lungs, again, you have your beta 2s, which are adrenergic. In the blood vessels, the adrenergics are the alpha 1s that mainly regulate what the contraction and the vasoconstriction of the vessel. And then remember that the muscarinic receptors in the heart, those cholinergic receptors are inhibitory. So that is why when acetylcholine is secreted on that SA node and AV node from the vagus nerve, which is the postganglionic piece, the postganglionic neuron of the vagus nerve, secretes acetylcholine on the muscarinic receptors on the SA node and AV node, and it's going to slow things down. Then we also have the baroreceptors in the heart, in the atria, that measure the amount of pressure. The parasympathetic, again, this is going to oppose sympathetic, so that means that it's going to inhibit sympathetic effects. And the acetylcholine hyperpolarizes by opening potassium channels in the cells and allowing potassium to leave. And so what that's doing is it's hyperpolarizing the actual cells. So when the heart is at rest, which is where we are right now in our rest and digest, it exhibits what we call vagal tone. And what that means is the brakes are keeping things in check, right? The heart's still beating, the heart's still moving, but it's not going very fast. Sympathetic, boom, right? We're going to increase heart rate, and that is going to be your norepinephrine, epinephrine, your dopamine. All those things are going to cause it to go up. It could be an emotional factor or a physical stressor or something that's being perceived. And then, of course, thyroid hormone can significantly increase heart rate because it's a sympathetic-like hormone. Glucagon will, remember, when you're in a sympathetic state, stimulate the liver to release glycogen to then increase blood glucose to be able to meet whatever that demand is of the sympathetic response. So your chronotropic agents, chrono means to count, chronotropic refers to heart rate. So when you increase heart rate, that means more sodium gates are opening and more sodium is rushing in. So what that's going to do is that it's going to increase the heart rate because it's a positive chronotropic agent. It speeds up the heart rate. Negative chronotropic agents are going to slow down heart rate. So for example, acetylcholine is going to be something that does that. The medication adenosine is going to be something that does that. So you're going to take a medication that directly impacts the heart rate response. What will happen when the heart rate decreases, okay, is that the heart would actually have to, in order to maintain cardiac output, you would have to have better contractility in order to get enough blood out for whatever things are going on. So one of the things that happens is that when you have an increased stroke volume, say you become much more aerobically fit, all right, your stroke volume is going to go up at rest because your heart is actually stronger. So you have an efficiency that has increased because now the heart doesn't have to contract as forcefully per beat to get the same volume of blood out. As you become more physically fit, what will also happen is that as your heart rate increases during a sympathetic response, you also become much more efficient with the stroke volume. Even though you're at a faster heart rate, your cardiac output also improves because the contractility in the heart increases because it becomes stronger. And these are things that happen in individuals who are becoming healthy. When you have a pathology related to that hypertrophy, that's different, right? That is not like, hey, I started doing cardio and now I got more fit. Usually when the pathology occurs in the actual heart muscle, it's because you have unhealthy resistance on the other side of that blood, and that means that now the heart is having to increase contractility. Then dromotropic agents, what they do is they increase the speed of the cardiac conduction. So the rate at which the SA node depolarizes, then goes to the AV node, then to the AV bundle, down the bundle branches, and then to the Purkinje fibers, anything that increases the speed of that cardiac conduction signal is going to be a positive dromotropic agent. Negative dromotropic agents, those are going to slow things down. So for example, adenosine impacts the SA node and the AV node. That would be a negative dromotropic agent. Then inotropic, this is going to refer to contractility. So this ties directly to what's going on with the calcium levels and how the heart cells are going to react to that. So when you have a positive inotropic agent, contractility is actually going to increase. And it increases because you get more calcium going into the cells, meaning the strength of contraction increases. So the hormones or neurotransmitters that are going to do this are going to be thyroxine, glucagon, and epinephrine, norepinephrine, and dopamine. But primarily, it's this epinephrine, norepinephrine, and the thyroid hormone that really have a significant positive effect on the inotropic function of the heart. Negative inotropic agents, those are going to decrease contractility. So if the heart becomes acidotic, that goes into anaerobic metabolism because there's a blockage of some sort, that would actually cause problems, and then that would increase extracellular potassium, or if an individual also is taking calcium channel blockers. So anything that slows down contractility is going to be a negative inotropic agent. This slide pretty much, as you guys saw earlier, it sums up what is taking place in the actual heart when you're going through the cardiac conduction cycle, right? So you've got your P wave, that atrial depolarization, then you've got your atrial systole, then the atria will repolarize and go into diastole while the ventricles are depolarizing here in that QRS. You're going to have the closing of those semilunar valves, and then you're going to have, after the depolarization of the ventricles, you have the repolarization phase following the systole that occurs between the S and the T, and then the ventricles will repolarize, and then we have that filling that takes place in between. So normal sinus rhythm is what you see here. So here's your P wave, QRS complex, it's narrow, and then followed by your T, then our little flat line, which is our passive filling, and then boom, we've got our next little P wave, QRS, and T. Then an easy way to start learning and kind of start understanding, well, how do you determine heart rate from looking just at the EKG paper? If you find a line right here, so your R is on one of the darker lines, right? This is basically 0. Then the next big box line, this is basically 300 beats per minute. Then the next one is 150, this one is 100, and the next one is 75. So on average, what you're looking at here is somebody who has 75, 77 beats per minute is what their heart rate is. And so what you're looking at here is basically a strip that is measuring 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. That's 2 seconds. Then as you go on. So this is a 6-second strip that is measuring heart rate. So you count these 1, 2, 3, 4, 5, 6, 7, and since this is 7 beats in 6 seconds, that means that you have 7 beats in times 10, which would be, gives you the full 60 seconds, and then that gives you a heart rate of 70. So on average, that is what you have here. All right, so 2 ways of doing that. Either you can look at it as the 6-second strip if you actually have a 6-second strip, or you can do what's called the quick and dirty way, which is going to be here, 300, 150, 100, 75. It's just inside that, so it gives you an idea that you're in the 70s in terms of the heart rate. Then we look at our P wave before the QRS. Yes, each QRS is identical. The increments between each is exactly the same. It's a regular beat, so this is a normal sinus rhythm. Oh, and then your PR interval is 0.12 to 0.20, so you look at it here. So it's no bigger, right? Here's our next one. So this is within normal limits, and it's a normal PR interval. Here's a sinus bradycardia. So this is going to tell you that this person's heart rate is actually slower than 60 beats per minute. So this right here is our 0, right? And here would be 300, 150, 100, 75, 60, and over here 50. So what is happening, 60. So what is happening here is that we actually have a heart rate that is actually less than 60 beats per minute. Or this is a 6-second strip. So if we times that by 10, right, that would give you 60 seconds. So we're looking at 1, 2, 3, 4. So on average, this person's heart rate is about 40 beats per minute. But we still have a sinus brady because we still have a P wave. So here's our P, QRS, and our T. Then sinus tachycardia, this tells you that the heart rate is going really fast. So here we have the 0, right? Then 300, this is 150, this would be 100. And so this mark right here is really close to the 150. So then we go through and we count our R's, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So this is about 120 beats in 1 minute. But because we still have our P wave, QRS, and a T, then everything is still within normal limits. Then a sinus arrest means that you dropped a beat, right? The sinus, right, the depolarization of the atria did not happen, which means that that also then did not conduct down to the ventricles. So what you're looking at here is P wave QRST, then we have our gap, which is our passive filling, P wave QRST, then we have a very long passive filling. So there was supposed to be a beat here, didn't happen, and then it picks up again. P QRST, gap, P QRST, and then it's probably going to do it again. So now what happens is you have before each QRS is identical, but now there's a pause, and then there's a new rhythm that begins after that. So this P here and P interval was okay, but from here, way over here, that was disturbed. So that is a sinus arrest. Then a PAC, that means a premature atrial contraction. And so here you have P wave QRST, normal, P wave QRST, normal, and then here it's starting to change a little bit. P wave QRST, and then boom, really fast, we had a P QRST. So this beat right here was premature. It came in earlier than it should have, but we still had a P QRST, so it's still a premature atrial contraction because you still had P wave QRS and T. Then what you are seeing here is called SVT, or supraventricular tachycardia. So here the rhythm origin is above the ventricles. So supra is above, ventricular is above the ventricle. So what happens here is that the heart rate is so fast that the P wave actually gets buried in the T, right? It's very difficult to see. But you see that the rhythm is still regular. It's just that it's going really, really fast. So when you look at this and you try to ascertain, okay, well, what is the heart rate? Remember, right here is where we start our count. This first line would be 300. This one is 150. So we are between 150 and 300 beats per minute, right? So you know it's going real quick. So it's over 150. So we count this, right? Our 6-second strip. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. So this is a heart rate of approximately 190 beats per minute. This would be a time where you would probably give something called adenosine, which is a medication that affects conduction through the, slows things down through the AV node. And what you're trying to do is to get that AV node to like put those brakes on and slow down what the SA node is telling it. Sometimes it works, sometimes it doesn't. And in some cases, you're going to have to actually do what's called cardioconvert if you can't get it to stop. And it's actually starting to affect the patient and they're starting to basically decompensate. And, or, you know, their blood pressure is starting to drop. Or you'll notice that their mentation is starting to suffer. And so what you would do is you would cardiovert this person by giving them a little jolt of electricity. And the one that's easy to remember for supraventricular tachycardia is 50. So you give SVT 50 joules, and it's pretty much like a little slap. Think of it as like a little bitch slap on the heart. It's like, just give them a whack, and you're trying to just like, bam, shock that little heart back into calming down. So that's really what this cardiovert thing is going to do. And you can also do it with ventricular tachycardia, so VTach with a pulse. If the person is unstable, that's when you would also do the cardiovert for that. And then the joules that we'd use for that are going to be a little bit higher. Then your AFib, this is when you have what we call a chunky and irregular. So this is irregularly irregular. So you can see that in between here, you've got the heart rate is kind of it's not regular. And then you notice that through here in the middle, it's kind of chunky, right? It's kind of bumpy. And so this is what's called atrial fibrillation. So what is happening is that the atria are going so fast that they're not even able to log like a normal P wave in here. And what's also occurring is that what's happening afterward, the QRSs are irregular because they don't really know when they're supposed to come. So they're still narrow, which is good, right? You still have a heart rate. And there's people that live in AFib their whole life, right? Or not their whole life. It usually starts when they're older. But in some cases, you may have to do something to convert this atrial fib, and you can also give a medication. Most likely the med you're going to give is going to be diltiazem. And that one tends to also help with this, but sometimes it's not going to resolve the arrhythmia. So one of the issues that can occur is that you can have these irregular heart rates, and then you'll have your AFib, and then all of a sudden you'll start having this really fast in between R waves. So you'll have, these guys will come in and they'll go boom, 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 and then it'll slow down again, and then it'll go boom, 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 boom again. And so that can be very dangerous for an individual when that is taking place. Then what you see here is just a number of different junctional rhythms. We've kind of already talked about them, but remember that a junctional rhythm, again, is one that does not have a P wave. So this would be much slower, so it's going to be between 40 to 60 beats per minute. In this case here, you'll see P waves, they call them retrograde, which means that they are upside down. So pretty much what happened is the signal went the opposite direction. And then over here, you have a junctional escape. So what this is showing is that it's a junctional rhythm, right? So there's no P wave coming in, and then basically the beat escapes is what they call it. So what happens is that we're going along, and then we had a junctional rhythm here, and then now the gap is much longer in between. So the whole even the junctional beat escaped. You skipped a junctional beat. Accelerated junctional is between 60 to 100. And here what's going on is it's junctional because there's no P wave, but now the beats are much faster, so they're coming together. Junctional tachycardia is going to be greater than 100 beats per minute, and in this case now the P wave is actually buried. It came after the QRS, and so because you didn't have one before that QRS, that's why this is a junctional rhythm, and then this one, it came late. So that's your junctional tachycardia. And then you can also have what's called the PJC, which is a premature junctional rhythm, which is similar to what I showed earlier. That can happen early, right? So here's your P wave QRST, P wave QRST, and then here's a junctional rhythm. Look at that, it came early. And it's junctional because it did not have an actual P wave. Then bundle branch blocks. This is what happened when you go to left and right bundle branch. And what you'll see are going to be these funky looking like little bunny ears. And what you're seeing is actually two R waves. And that is indicative that the ventricles did not beat at the same time. So the ventricle that depolarized first is right here. And then the one that depolarized second is right here. And so what happens when you see these, you'll actually do your 12 lead. And then you look at your V1 and your V2. And then the actual direction of that QRS complex and what it's doing is going to tell you if it's on the left or if it's on the right. So in looking at this one, in your V1, V2, it's pointing down. So think of it as your blinker. When you click down, you're going to go left. So this would be a left bundle branch block. If it was going up, then it would be a right bundle branch block. In this case, if I'd done a 12 lead and I looked at my V1, V2, then it would be indicative that the blockage was actually going to the right. So that's how you can tell when you're looking at your bundle branches. And you guys will learn when you're doing ECG, all the little tricks for this. Then heart blocks have to do with now the signal is actually being affected in terms of conducting from that atrial down to the actual QRS. And so what happens here in this first degree heart block, here's your P wave, and then it goes, and then here's your QRS. But you notice that the distance between the P and the Q are very, very far apart. They're much greater than 0.20, which is what your normal limits are. And so this is where you're seeing that our wave is much longer. So this would be a first degree block. Then we have what's called the second degree block type 1, and this is where you have an increasing distance, so it progressively gets longer between the P and that R. So here is your first one, and then you're going to your next beat. Now it got longer, now it got longer, and then what happens is you drop a QRS. You get your P, but it drops off the QRS, so it never conducted down to the ventricles. So the ventricles here never depolarized, there was never systole, there was no contraction, so the blood just sat there. Then the second degree heart block, the type 2, which is called Mobitz type 2, the one I just talked about, the type 1 is called the Mobitz type 1 or a Wenkenbach. Don't ask me why they named them these things. Probably the dudes that decided to, hey, this is what we're going to call it. So here's your second degree. So in this one, what happens is you have a fixed PR interval. So the distance between the P and the Q is fixed, right? But what happens is that eventually you're going to drop off a QRS. So here's your P wave, QRS, T, right? P wave, QRS, T. So these intervals are normal. And then here's that P wave, and then boom, it drops off. So the difference between a second degree type 1 and a second degree type 2 is that in the second degree type 1, the interval, the PR interval, gets progressively longer, and then you drop off. In the second degree heart block type 2, the PR interval is fixed, it doesn't change, but you eventually will drop off a QRS. And then here where you have the third degree heart block, now what's happening is you have your P waves. So here's a P wave, QRS, here's your T. Then you have your P wave. There is no QRS immediately following that, but it happens later. And then you're coming through, here's your T, then here's a P, and then there's another big gap. And so what's happening here is the P waves are beating on their own. So your atrial depolarizing and causing contraction, but the ventricles aren't listening. The ventricles are doing their own thing. And so you'll have regular intervals between the P's, and you'll have regular intervals between the T waves here and here. So this is your regular P wave interval to this one, and then your actual R's here to here, those are going to be normal. So it's really weird how this ends up happening, but we have a real problem because the P waves are completely unrelated to the QRS that's happening, and now you have atria doing their own thing, ventricles doing their own thing, and you can see that this person, when you have a third-degree heart block, becomes very bradycardic. Same over here with the second-degree heart block, they will become bradycardic. And so this is why these types of heart blocks can be so dangerous. So again, I've got all the words, right, and all the slides for you guys to go through, and you can see those obviously in the PowerPoint handout. But the types of symptoms that people are going to have with a first-degree heart block, usually not much, right, pretty much asymptomatic. Sometimes with the second-degree type 1, they'll have palpitations. With the second-degree type 2, they, again, will have palpitations most likely. Most often what the solution is for this is a pacemaker. And then the third-degree heart block, they will definitely they'll be reporting that they feel like they're fainting or they did faint. That's what made you guys come respond. And in some cases, they're going to have to do a pacemaker to fix that. Then a PVC is a premature ventricular contraction. So here's a normal beat in the middle, right? P, QRST, P, QRST, and then all of a sudden, the ventricle just kind of jumps in ahead. There's no real P wave, and goes blah. It's like a drunk ventricle, all right? That's kind of what happens. So here's a PVC, and here's a PVC. And how you know it's a PVC is the QRS is really wide, and there really is no, true actual T wave that you actually see. Then the ventricular tachycardia, these are going to be wide, because you can see here is the ventricle, the origin of the actual rhythm is from the ventricles, and you know that because this QRS is really wide. And so here's your ventricular rhythm. At least this one is what we call monomorphic, which means it's regular. So people that are having this are probably going to have a pulse. Sometimes they don't. But if you have VTach with a pulse and the person is stable, you can actually, just try vagal maneuvers to calm it down. But sometimes this is obviously going to lead into pulseless VTach, and that is one of the rhythms that you can actually shock. If you have VTach and it's a little bit more narrow or it looks like this, you can try adenosine, but sometimes that's not going to work. And then when you have VFib, this person's pretty much dead. This is a shockable rhythm. So VTach with a pulse and VFib are your two shockable rhythms. Sometimes you'll have electrical activity that looks like they're having something, but you don't feel a pulse. That's called PEA, which means pulseless electrical activity. And so you can't shock that because there isn't anything to work with. There's absolutely no real, true electrical activity. And then this is just another example of what VFib looks like. So here probably the person was doing okay, and then all of a sudden they just went into, fibrillation. And so what's happening here is the heart rate is going super, super fast. Like there's no organized rhythm whatsoever. And there's no P wave, there's no real QRS, it's just a mess. So extremely irregular. And then this is your asystole. So this is your flat line. Again, this is not a shockable rhythm. And for this one, you just do your best, right? You want to try and get them back. So you're doing your CPR, you're pushing your EPIs, you're not able to shock, obviously, but hopefully at some point you'll get a shockable rhythm from the epinephrine that maybe kind of wakes up a few cells, but it just depends. So asystole is asystole. And when you do this, you want to make sure that you've got asystole. If you're going to call somebody dead, then they need to have it asystole in two contiguous leads. Then your ST elevation, we talked about that earlier. So remember that you're going to have ST elevation in the leads of the region of the heart. So this would be that right marginal artery. And so what you're looking at here is, here's your ST elevation, obviously, the S and the T. You could have a little concave here. And then what will happen is in your reciprocal leads, which is 2,3 AVF, then your reciprocals are going to be 1 AVL. This shows that you have a little bit of that ST depression here taking place, and then you can actually see that. So when you look down here, here is your, 3 obviously lead AVF. Here's your tombstone. We also call it that. And then over here in your V2, you also have an ST depression. Here in lead 1, you have an ST depression. So anyway, so you guys will learn all about this when you get to the actual EKGs. But this person is not having a good day because 2, 3 AVF, lots of ST elevation, and then lead 1, AVL, V2, V3. Here you've got depression. So this is telling you that there's bad things going on within that left ventricle, and they're not doing well. All right, so here are your MIs, your myocardial infarctions, where they happen and where you would see it. So any anterior one that's going to involve the LAD, that's where you're going to have your V1, V2, V3, V4, and this is your left coronary LAD. Common dysrhythmias that can cause this would be AFibs, aflutters, tachydysrhythmias, right? So it's going too fast, PVCs, second degree AV blocks. Then the lateral side, which involves the circumflex, this is now you're looking at 1 AVL and V5, V6. You're probably going to see initially some PVCs with this. And then septal, that is going to be your LAD again, V1, V2. Second degree heart blocks and third degree heart blocks will be common dysrhythmias that cause this. Then your inferior, this is going to be the right coronary artery going along that region, which then feeds into the right marginal. And then this is your 2, 3 AVF. Sinus brady, sinus arrest, first degree and second degree blocks, and your PVCs, common for these. And then the right ventricular infarct, this is where you would then take your V4 and move it over to the right side in the same spot that V4 is on the left. And then that would show you if there's an actual issue going on the right side of the heart, and that would confirm a marginal branch issue. You could also move V5, V6 over to the other side if you really want to really make sure if that's the case. And then you can add leads if you want to see going around the back to get a little bit better view. But atrial flutter, atrial fib, AV blocks, and PACs would cause the issues for the right ventricular. And then the posterior part of the heart, this is where you would go around to the back and add leads, V7, V8, V9. And then that's going to explain that's could be the right coronary artery that then loops around the back, or the circumflex, as you know, loops around the left and then meets up in the middle and sometimes feeds that posterior part. So that is where if you have SA or AV node blocks, that could cause that particular dysrhythmia. So we've talked a lot about dysrhythmias. We've talked about what a normal EKG is. Now we're going to get into the actual issue that happens when your perfusion triangle suffers. So we have here the heart, the blood, and the vessels. So the heart is your pump. The blood vessels are the pipes that contain the blood, right? You don't want leaks. And the blood itself is the actual fluid in the pipes. So your cardiac output is going to be directly influenced by how much blood comes back, right? Because we have to have a pressure in those vessels to return that blood back to the heart. And then in order for the blood to get into the vessels, you have to have a pump that is working. And then, of course, you have to have blood. And in the blood, we have plasma fluid. And that plasma fluid is very important because that ties into your volume. So we're going to get into how it is that blood pressure works. What I first wanted to show you here was basically the main subdivisions of what comes off the aorta. So here's your heart, here's your aortic arch, and you know that the right side here, this is the brachiocephalic trunk that will subdivide into the right common carotid and the right subclavian. In the middle, you have the left common carotid, and then you have the left subclavian. So those are the main arches coming up off the aortic arch. Then that chest, right, it's going to come down behind the heart, that is your thoracic aorta, and then it's going to go down into the abdomen and then down into the legs. So the branches that are going to come off that right subclavian, first most important one is going to be that one that leads into the vertebral arteries that are going to then feed the posterior part of the occipital lobe, the brainstem, and then feed into that circle of Willis or that cerebral circle in the middle. Then the subclavian will subdivide into the axillary, then into the brachial, and then go into the radial and the ulnar, and then into the arteries that are obviously going to feed into the hand. So when you're taking a pulse on the wrist, you're actually measuring the pulse rate at the radial artery. If you can't feel a radial pulse, say you go up and you try and find a brachial pulse, can't feel that. Let's hope there's one at the carotid, right? So these out here are considered peripheral pulses, and then the carotid is going to be your central pulse. You can also listen here, obviously on the heart, to listen if there's actually anything beating. Then the aorta itself, when it descends down through the abdomen, right, so it's going to cross through the diaphragm, this first branch here is called the celiac trunk. And the celiac trunk, the artery on the right, going this way, is the right common hepatic. So this is going to go directly into the liver. Then you have the gastric artery that is going to go up into this, is called the left gastric, that goes up into the stomach. And then this one here, this branch goes over and feeds into the spleen, so that's the splenic artery. Then you drop down a little bit more, and now we're going to have the superior mesenteric, and then you have the inferior mesenteric. Those are going to feed the blood directly into the intestines. Then you have the renal arteries, which go into the kidneys, the left and right kidney. Then just slightly below where the belly button is, that is where the aorta splits, and then it becomes the common iliac arteries. These will then become the internal iliac and the external iliac, and as it enters into the leg, then it becomes the femoral artery. And then these will actually subdivide and become the tibial and the fibular. And granted, there's a few other ones that it divides into, but these are basically the main primary branches of what you're primarily seeing here on this image. And then obviously if the person has a pulse at their feet, which is always a good thing, that means the blood is going down there. So when you check a pedal pulse, you're actually checking here as it's coming around the metatarsals, just anterior to where the tarsals are. And then if you're checking to see a pulse behind that tibia, you look at that tibial pulse, which is just posterior. You can also find a pulse here in that dorsal pedis artery. Then what you see here, obviously these are the veins, and then this is the venous circulation. So we'll have the veins that obviously come down from the head. Again, here's that vertebral vein, and then you're going to have your jugular veins. These are going to empty into that subclavian vein and then go in at the little curve right here. This is the brachiocephalic, and this is also the brachiocephalic. And then those together enter into the superior vena cava that empty into the heart. Then the inferior vena cava, you can see that's going to bring the blood from below the diaphragm. That includes the blood from the legs, and then obviously the pelvic region, the abdomen, and then up into the heart. And then our veins that are in our arms, also those are going to return back, get into that subclavian, and then obviously join into that brachiocephalic region. So for our purposes, obviously the veins that I want to kind of point out here that are going to matter to you guys is this one right here. This median cubital vein is basically your antecubital vein is also the other word for it. This is your money vein in terms of if you're having a hard time getting an IV. Ideally, what you want to do is you want to try and get an IV in this vein, which is your cephalic vein that runs down and then is parallel along that portion of the arm. The radial vein is actually a little bit deeper, and so is the ulnar vein. But this cephalic vein that is running through the side where the radius would be, this is more on the surface, and this is one that you can usually see pretty well on individuals once you do the actual tourniquet up here. If you're trying to get a lower vein, do the tourniquet further down because it's going to give you a little bit more pressure down in this area. And then on the posterior portion, you can't see the posterior of the hand here, but there are some really good veins in there as well. And so it really helps to kind of learn, okay, where is like in general the anatomy for the veins if you do end up needing to do a difficult IV. And then obviously the feet are always going to have a lot of really good veins in there. But if you're trying to aim for a good one, this one right here where the dorsal metatarsal vein is that leads back up into what's called the saphenous vein, this is a good one too, because this was going to lead directly into that into the external iliac vein that then feeds back into the inferior vena cava. But the closer you are to the heart, the better off you're going to be in terms of sending in the fluid that you need or the medication. Then in terms of what blood pressure is the force per unit area, so how much pressure is being exerted on the sides of that vessel wall. The pressure entering into the vessel is always going to be higher than the pressure leaving the vessel. So if you remember proximal to distal, all right, here, this is going to be your pressure gradient, right? And this is the driving force that keeps the pressure moving because it's coming from a higher to a lower. So just like what you learned in diffusion, that is how the pressure is going to move. And then we measure blood pressure, obviously the systemic arterial circulation, and we're measuring the pressure in the brachial artery because it's the closest and least invasive way to be able to get a blood pressure of what getting the estimate is for the systolic from the heart. Blood flow is the volume of blood moving through a vessel, an organ, or obviously your entire circulation, which is your cardiac output. So it's going to be equivalent to what the cardiac output is. It's relatively constant at rest, and when you're at rest, it's mostly being directed, obviously, always to our brain, always to our heart. It's going out to all the tissues, but it's mainly focused in that gut and kidney area. Now, when you have to shift the blood back and forth, depending on what you're in, whether it's sympathetic or parasympathetic, that is when blood gets shunted from some areas to others, depending on what you need to do. So each organ that we have, our heart, our brain, our kidneys, our adrenal glands, our pancreas, right? They each have their own minimum milliliters of blood that they can get. And they have to have a minimum pressure. And so that's called perfusion pressure. What that means is if you have perfusion pressure, you need that blood. It has enough pressure to get to the organ and meet that organ's needs. So when we look at pressure, there's this equation that means flow is equal to delta P. Delta P is the difference in pressure, P1 minus P2, right? At the beginning and at the end of that vessel. And then it's divided by the resistance. So what this is telling you is that if you increase the pressure gradient, so if it gets bigger, then the flow rate is going to also increase. If it decreases, then that flow is going to decrease. So what you see over here is here at the beginning, we're at 50, right? And then from here, we're going to move to where it's at 10. So here, your delta P, your difference, right? Your pressure gradient is at 40. Now we move to another vessel. Now what we did is we increased the actual P, right? So this is 90 now, and then at the end it's 10. So now the delta P went to 80. So in here, now we have double, right, the amount of flow that we had in the first vessel because it increased. So if the pressure gradient increases, then the flow is going to increase. So essentially what you want to think of is basically your pump helped increase pressure. So now you have more flow of the blood going through. Your pump is pumping more, right? And then what also happens is that if for some reason you have an increase in pressure here and an increase in pressure at the end, right? Now what's going on is you got to look at, okay, what is our difference here? So what happens here is you could have here it's 90 to 10, right? And here's 180 to 100. The difference is still 80, right? So what has happened here, even though the larger absolute values are bigger, your pressure gradient still remains the same. Even though the P1 and P2 increased, you still have the same pressure gradient. So it doesn't always mean that just because your pressure gradient at the beginning, right, your P1, your P2 go up, it's what the difference between those two that ends up mattering. So what I've written here is that the blood flow is directly proportional to blood pressure gradient. So if the blood pressure gradient increases, blood flow is going to increase, right? Then with resistance, it's different. So if resistance increases, that is your opposition to flow. So if resistance goes down, then that means blood flow will go up. And if resistance goes up, then blood flow is going to go down. So all you're looking at in here is the diameter of the actual vessel. So what happens over here on the right, here's your blood flow, right? And as the blood is flowing through, you always have some kind of resistance. So for example, shorter vessels are going to have less resistance. Longer vessels will end up having more. In your vessel radius, if the vessel radius or the diameter is bigger, you can fit more blood in there, so you have more blood. If the vessel radius is smaller, you can fit less blood, and therefore that means the resistance went up, and now you have less blood being able to pass through. If viscosity increases, then that would mean that the blood is thicker, and that would cause an increase in resistance, like what happened here. If you decrease viscosity, then that makes the blood thinner, and it decreases the resistance. So the most adjustable variable that we actually have in dealing with resistance is going to be the vessel radius or the vessel diameter. Because what that comes down to is that we can very quickly change how vasoconstricted or how vasodilated a vessel gets based on what the sympathetic response is. And again, remember that the sympathetic system is what innervates our systemic vessels, and those are what are going to regulate constriction. So if you go into a sympathetic response, you're going to get an increase in pressure. But remember that what also happens is that the pump, the heart, is also going to be contracting more forcefully and beating more frequently. So your pump pressure also goes up. And that is why we can still maintain flow. If we didn't have that ability, then we would be in trouble. So we need to be able to have that. But just remember, if the vessel dilates, then that increases blood flow, decreases resistance. If the vessel constricts, then that decreases blood flow because resistance went up. Then when you look at your vessels themselves, again, this is it, easily showing what I just talked about. Here is your vessel itself, right? Same pressure gradient, but if for some reason the vessel, right, so pretend that these two are the exact same vessel. Here's the vessel dilated, and then if for some reason, boom, the resistance goes up, then we constrict the vessel, then what's going to happen is you're going to decrease blood flow because there's less room in the actual vessel. And so resistance is inversely related to the actual ability for the blood to flow. And so we need to have a means to be able to regulate that. So the only way to maintain, like say here, this was your flow rate. The only way to maintain the flow rate here when the vessel constricts that you had here in the dilated vessel would be to increase the pump. Otherwise, you're going to decrease blood flow through a constricted vessel. All right? Here again, you can kind of see a little bit of what happens. So here's your healthy artery, right? And say now you're starting to have occlusion with plaque buildup. So look what happens when you increase just 20%, right? So your vessel is now obstructed 20%. You would need, in order to maintain the same flow rate that you had up here in that healthy artery, you went from 120 as a normal pressure rate, right? So say this is our systolic 120. Now it turns into 293 that you would need to pump in the same way. Now, granted, this is a very extreme example, but you can actually significantly decrease what that flow rate is through the vessel when that resistance increases. So that's why it's important to do our best to try and maintain healthy arteries. So if resistance goes up and you want to maintain the same flow rate, you're going to have to increase your pump pressure. If you don't do anything with the pump pressure and you increase resistance, then flow is going to decrease. Then the way most of our arteries are set up is that with this, for our capillary networks, we have this parallel resistance map that's going in with the plumbing. So pretend this is our aorta, and then it's breaking up into, let's just call this the kidney, right? So you have your two kidneys. So now it's going into the left and right kidney. And then once it gets into the kidneys, it starts going into smaller and smaller arterioles until you get down into the capillary network, where you now have what we call series resistance. And so you'll have arterioles that will branch into the capillary bed, and then we'll pick up the blood flow at the venous end. So here you can actually distribute the blood into your capillary beds, oxygenate the tissues, and then off it goes. So this is called the parallel resistance, and this is called the series resistance. Don't worry, I'm not going to ask you about that on the test. Now, in terms of how our blood pressure is set up, we have blood pressure categories. It used to be that 120/80 was our normal, 100/60 was your baseline for low normal, and anything below that was hypotensive. And then 140/90 was high, and anything above that would be hypertension. But now we have these different grades of what we call stages. So normal is still a systolic 120 and a diastolic less than 180. So 120/80 is still normal. Elevated is 120 to 129, and then diastolic still less than 80. All right. But now when you start getting into the stage 1 hypertension, what we're really looking at is this right here. The diastolic pressure is a big deal because this is telling us just how much resistance there is in the systemic circulation. So how much resistance does your left ventricle have to pump against in those vessels in the systemic circulation? So as this starts to increase, what is going to happen is now your systolic pressure is going to have to increase, because if you want to maintain that flow rate, remember the pump has to pump harder. So this is what they refer to as a stage 1 hypertension. Then when you get to stage 2, now you're looking into that 140 or higher for systolic, or the 90 or higher. So this would be where it used to be just high blood pressure. And now when you get into what we call a hypertensive crisis, this is an immediate, either they call 911, right? Or they hopefully go to their doctor. But this would not be something that I would want to leave in terms of a high-risk refusal, just because of the risk of what can happen in the brain and how the brain is going to react to this perfusion that is happening at a very high pressure. So 180 systolic and or 120 for diastolic. So this 180 over 120 is very, very dangerous. Then remember that systolic is the pressure exerted when you have ventricular systole, when the heart contracts, and then diastolic pressure is the lowest level of arterial pressure, which is when the heart is in diastole. Then your pulse pressure, this is a very important thing that you need to understand. This is systolic minus diastolic. So obviously this is part of that pressure gradient that we talked about earlier. And so it's very important that our pulse pressure, right, that there be a difference here. You never want your diastolic to be bigger than systolic, because that means your blood is not flowing, right? So normal pulse pressure is going to be between 40 to 50 millimeters of mercury. As your pulse pressure widens, sometimes it's going to be indicative of what we call brain swelling. So you're having some kind of cerebral edema, intracranial pressure is actually increasing, something is going on that is causing brain to be squished. So now we start getting this widened pulse pressure. A narrowed pulse pressure would be when you have less than 40 millimeter of mercury difference between the two. And this is indicative of when cardiac tamponade, for example, it makes it much harder for the heart to contract, something is squishing it, or now a person is starting to go into some kind of shock, right? So now the difference is becoming much, much narrower. Mean arterial pressure is a function of your pulse pressure. So it's equal to diastolic pressure plus 1/3 of the pulse pressure. So 1/3 of the difference between systolic and diastolic. Normal MAP is between 85 to 95. If you have a MAP that's less than 60, and this is a huge, super important number, right, your brain is going to suffer, all right? Ischemia, organ failure. This is when you can start going into that issue. So you want your MAP to be greater than 60 in order for your brain to be properly perfused. A MAP that's too high, greater than 150, this is now going to cause cerebral edema. So you have this kind of little range in terms of how much can the brain handle, all right? And the perfusion pressure that the brain can handle is between 50 to 150. That is its happy place. That is a little bit different. That's called cerebral perfusion pressure. That is a little different than this MAP. This is just your overall mean arterial pressure that includes the entire body. And again, the big number here is that your minimum is going to be 60. What happens with MAP and with your systolic pressure is obviously the further away you get from the heart, it's going to continue to decrease. It'll be highest, closest to the aorta, and then lowest, obviously, in the capillary level. So when you get into this hypertensive crisis and you start looking at what is going on with our pressures, we still have, right, 180 over 120, we have a systolic minus diastolic pulse pressure of 60, but what's going to be affected here also is going to be what happens with the MAP. So here's our MAP, right? Decreases the further away it gets from the actual aorta. And then obviously down here when we get into the veins, we don't have an arterial pressure in the veins because we're now in the veins. So what's going to happen here is as we return back to the heart, venous pressure will gradually increase. Then remember that we have that closed circuit. So the pressure down here at the capillary level is going to be at the lowest. And then as we start picking up through the venous circulation, it's going to increase the closer it gets to the heart. But our highest pressure, the highest systolic pressure, is always going to be because of that left ventricle pumping the blood out into the systolic or into the systemic circulation. So the arterial smooth muscle, I mentioned this earlier, is innervated by the sympathetic system. So when you go into a sympathetic response, you're going to get generalized vasoconstriction. And so normally what that would do is it would decrease the volume of blood, right, that's passing through the vessel because the resistance has increased. But what also is going to happen is that it's going to increase your systemic blood pressure. Both diastolic will go up and so will systolic. However, the systolic pressure, because of going into a sympathetic state, is able to increase significantly enough to overcome the difference of the increase that's happening in the diastolic pressure. So for example, when you work out, your systolic blood pressure can go up into the 180, 190, but your diastolic is going to be maybe, say it's normal at 80. It will increase about 10 to 15 millimeters of mercury. So it's not going to go up that high. So you still have a nice difference in terms of your P1 to your P2. When we work out, we are meeting a need, right? Our skeletal muscle needs to have the right amount of blood flow. And we take blood from the gut and the kidney and then shunt it over to that skeletal muscle, and that also goes to the skin to help us cool off. The perfusion that is happening in our brain remains the same. It stays constant, right? We're not going to get this sudden huge increase of blood that's going into our brain because that could cause other problems. Then when your sympathetic response is done, now what's going to happen? Your parasympathetic takes over, and when the parasympathetic is in charge, that means it's inhibiting sympathetic. So what's going to happen is you're going to have more vasodilation because of the parasympathetic response, but it's not that the parasympathetic system has nerves going to those vessels that are innervated by the sympathetic. That's not the case. It's that the parasympathetic is now inhibiting sympathetic. And so therefore, because sympathetic is inhibited, that causes vasodilation. And what I want you to understand is that we always have a little bit of sympathetic tone, meaning that there's always a signal going to our vessels. Because if we had zero sympathetic tone, if it completely turned off, then that means that our vessels would completely collapse and just sit there, and we would have this pool of blood that didn't have a pressure gradient. So we always have a little bit of sympathetic tone in our vessels, no matter when we are in that parasympathetic state. The precapillary sphincters are what regulate, obviously, blood flow into the capillary. And so that is where your pressure, obviously, is going to be coming down the lowest. But one of the things that can happen is that you can get this increased blood flow, where you get this greater vasodilation when you have some kind of inflammatory response. So we have tissues that have that contain mast cells, and mast cells have histamine. And histamine is a very, very powerful vasodilator that opens up our vessels and allows increased blood flow to go to the tissues. And so what happens here is that sometimes you can end up getting too much fluid blood going to the region, and then you get seeping of the plasma fluid going out because the hydrostatic pressure in the vessel is so great, and then we aren't able to get enough of it back in, and then you can go into shock. So you don't want that to happen. When you get a severe vasodilation, that's when you go into shock, and that's when you start having that fluid leak into the tissues, and the person results with edema. So this can happen in the peripheral circulation, and it can also happen in the brain. So here's that capillary bed. So here's your arterial side, here's your venous side, right? The precapillary sphincters, these little guys, are what regulate what's going to come in. Remember that the regulation is based on what is the chemical environment that the tissue is in. So if the tissue is more active, you're going to have dilation of these precapillary sphincters, and more blood is going to flow into the capillary bed to make sure that the tissue can be oxygenated and carbon dioxide can be cleared out. If the tissue is not as metabolically active, there's decreased blood flow into the capillary bed, and then that blood gets shunted through and goes into that venous side. So how this is regulated is what's happening at the capillary bed based on the hydrostatic pressure and what we call osmotic pressure. So here, when you're looking at these words, this is where the fluid pressure, right, in the arterial end basically pushes fluid out. You have hydrostatic pressure that is pushing fluid out into that interstitial space. And the hydrostatic pressure is going to be greater than osmotic pressure. The osmotic pressure is actually the pressure that is exerted by the concentration of the solutes in the venule that create the pressure gradient. So remember, what osmotic pressure means is if osmotic pressure is high, then that means there's more solute, less water. And so what you know about osmosis is when there's less water, the water is going to move toward where it's lower, right? So that means that if the fluid is outside greater, the hydrostatic pressure outside, then it's going to move in to where the osmotic pressure is basically causing it to pull in. So let me show you on here. So here's your arterial pressure, all right? Comes in, hydrostatic pressure here dominates because you have more pressure, more fluid coming in, and you also have that pump pressure, right, from the heart. So what happens is the fluid pressure is greater, there's more fluid in here, so more fluid is going to flow from the vessel out into the interstitial space. Now, the fluid, the blood, right, is going to continue to flow in the capillary, and then on the venous side of the vessel, now what happens is that there was more fluid out here, less fluid in here, because remember we pushed more fluid out, and so what happens is in here, the osmotic pressure is greater because we have more solute inside, less water. So what happens is the fluid is going to flow from out here back in. And so what happens when it says the osmotic pressure is greater on the venous side, it means that the fluid is going to get pulled in. So osmotic pressure means pulling in fluid, right? Or fluid is moving from where it's in higher concentration out here, pulling back in. And it was in higher concentration out here because it got higher concentration here when it got pushed out of the actual capillary. So when I ask you in the test and on the quiz about arterial pressure, hydrostatic pressure dominates on the arterial end, and on the venous end, it's osmotic pressure that dominates. So on the arterial end, fluid is going to go from the vessel out, and on the venous end, fluid is going to go from outside the vessel back in to the vessel. And why this happens is because we have a protein in our blood that's very important. It's called albumin, and what albumin does is that it helps provide that osmotic pressure. So it provides that solute that is greater inside the vessel, which causes fluid to be less inside the vessel on the venous side, and then fluid is going to move from outside the vessel into the vessel. And so that's how we maintain our capillary gradients. Then our respiratory pump, our muscular pump, and the actual vasoconstriction of veins under the sympathetic control help with the venous return that happens from our systemic venous circulation. So the lungs, when you breathe in, right, you squish down, you create pressure on the vena cava. Then when you exhale, now it relieves that pressure and fluid, right, the blood continues to flow back to the heart. So that's what we're doing up in the chest. Then in the periphery as well, we have a muscular pump. When your skeletal muscle is active, it is actually creating pressure from the skeletal muscle onto the veins and helps push the blood through. And then there's valves that help prevent the backflow. And then the veins themselves, remember, are under sympathetic control, which have a pressure gradient. So right here, from our venules and our capillaries, right, there's no more arterial pressure. So now the blood has to return back to the heart. And the only way to do that is to have a venous pressure. So the venous pressure happens because we have muscle, right, that is squishing, providing pressure against these little vessels that help bring it back. We have the lungs that when you exhale out, you relieve pressure in the chest cavity, and then that allows for blood to continue to flow back into the heart. And then you have the sympathetic innervation that helps provide a pressure gradient in the veins, where what happens is it's going to start out a little bit lower here, and then gradually, as we get to the bigger ones, it's going to continue to increase and then dump into the heart. So remember that our vagus nerve, parasympathetic, and then within our actual medulla oblongata, we have our cardioinhibitory and cardioacceleratory centers. So the vagus nerve is cardioinhibitory, less contractility, less beats per minute, right? Which also then is going to decrease, obviously, our stroke volume. Cardioacceleratory, sympathetic. So that is going to increase heart rate, cardiac conduction signal, and contractility. So what happens with the vasomotor center? This is also located in our medulla oblongata. And this is what's going to regulate systemic vessel diameter. So what's going on in an arterial network, and then what's going to happen in our venous network. So you want to primarily think about the arteries, right? They have a lot more muscle in the actual tunica media, which is that middle layer of muscle. And so this maintains that vasomotor tone. So it's our sympathetic system that maintains the vasomotor tone in our systemic circulation. And it knows what to do based on what is happening from baroreceptors and chemoreceptors. We have baroreceptors, which measure pressure in our carotids, in our aorta, in our atria, in our kidneys. They know how much pressure is passing through. So that information gets relayed and then is regulated based on what the vasomotor center is going to do coming from the medulla oblongata. So if your vasomotor center is inhibited, what that means is your parasympathetic system is in charge. So you're going to be in more of a vasodilated state, which means the pressure in the vessel is going to be less. If you stimulate the vasomotor center, now you're in a sympathetic state. So now what's going to happen is you're going to have more vasoconstriction. Pressure is going to increase because you've vasoconstricted that vessel. And remember that in a sympathetic state, your pump, the heart, is also beating more frequently and contracting more forcefully, which is how we're able to maintain that pressure gradient through the vessels even though there is more vasoconstriction. So when your cardioacceleratory center kicks on, you are in a sympathetic state. So what that means is that you have stimulated dromotropic, chronotropic, and inotropic. The signal goes faster, the heart beats faster, and contracts more forcefully. So pressure exiting the heart goes up. You also, at the same time, right, when you're in a sympathetic state, you are inhibiting parasympathetic, which means that you are not slowing things down. But if you inhibit sympathetic, that means that when you inhibit sympathetic, that means the dromotropic slows down, which is the signal, chronotropic decreases, heart rate goes down, and inotropic means the contractility of the heart decreases. So what has happened here is if you inhibit the sympathetic response, things here become parasympathetic. If you stimulate the sympathetic response, we're going to have a sympathetic reaction in the signal, the heart rate, and the contractility. Then, if you stimulate cardioinhibitory, that means you are turning on parasympathetic. And if parasympathetic is on, that means that you are inhibiting your sympathetic. So, if we stimulate parasympathetic, that is dromotropic, slow down the actual signal. Inhibit chronotropic, decrease heart rate. Heart cells depolarize less frequently. And we also decrease contractility. Now, if we inhibit parasympathetic, that means you are now kicking on a sympathetic. So that means that you would inhibit parasympathetic, and if you inhibit parasympathetic, it's the exact same thing as saying stimulate sympathetic. So if you inhibit parasympathetic, which means we are turning on sympathetic, now you have this sympathetic response here of speeding up the signal, increasing heart rate, and increasing the force of contraction. So obviously these two are opposites. Then your baroreceptors, the way they're going to respond is to a decrease in pressure, right? So they're going to stimulate the vasomotor if there's a decrease in pressure and they don't feel the stimulus. So we're going to kick on a sympathetic response. So that means vasomotor is stimulated, cardioacceleratory is stimulated, and we inhibit the cardioinhibitory, which is your sympathetic. Now, if there's too much pressure and we need to correct that, now we're going to turn on parasympathetic and inhibit sympathetic. So we inhibit the vasomotor, which is sympathetic, we inhibit the cardioacceleratory, which is sympathetic, and we stimulate the cardioinhibitory, which is parasympathetic. So when you look at this image, this is just giving you that seesaw of how does this get regulated. So increased pressure will be felt by that sinus that is in the carotid arteries. It sends the signal to the brain to say, hey, stimulate the cardioinhibitory, inhibit cardioacceleratory, and inhibit vasomotor. So this causes that inhibition of the sympathetic response, and heart rate decreases, contractility decreases, and we decrease the cardiac conduction, plus we vasodilate the vessels. So that causes a decrease in cardiac output and a decrease in resistance, which returns blood pressure back to normal. Then if we get too low of a pressure, now that's going to cause a sympathetic response because we feel no pressure here in the carotid arteries. Then that signal goes to the brainstem and boom, we kick on the sympathetic response. So now we stimulate our cardioacceleratory, we stimulate vasomotor, and we increase our cardiac output, right? So then that increases blood pressure and then helps return blood pressure back to normal. Then the vasoconstricting chemicals, just to remind you guys, these are your sympathetics. So it's going to be your norepinephrine, epinephrine, and your dopamine. And then again, remember that angiotensin II is produced by the renin-angiotensin mechanism or renin-angiotensin system, which causes angiotensin II to be produced. So angiotensin II is a generalized vasoconstrictor and also triggers aldosterone secretion so that you will reabsorb more sodium. And when you reabsorb more sodium, you also absorb more water, so that increases volume. So then ADH, antidiuretic hormone, also is a generalized vasoconstrictor, increases pressure, and increases water reabsorption. So all of these together are working to increase blood pressure. Then when you need to decrease, now we kick on parasympathetic, which inhibits sympathetic, and it inhibits the secretion of your norepinephrine, epinephrine, your dopamine, your ADH, inhibits the renin-angiotensin system, which produces the angiotensin II, which now causes inhibition of angiotensin II, and also inhibits aldosterone because angiotensin II would be one of the things that stimulates aldosterone secretion. Plus, we secrete ANP, which remember means you need to pee, and this is your atrial natriuretic peptide, which is a generalized vasodilator, and it's also a diuretic. So it's going to help decrease blood volume by causing you to pee out more sodium, and then you keep more potassium. Then the velocity of your blood flow is what we call inversely related to the total cross-sectional area. So the more cross-sectional area there is, so just think, the more parking lots there are, right? That is going to cause for that blood to be able to flow much more easily into the space, right? So the velocity of the blood as it flows into the parking lot goes slower, and it's going to be much faster in the aorta where there is a lot less space, right? So think of this as what happens in your streets. Here you are on the 101 or the 202, right? You're able to go super, super fast. But now we get to the side streets. Boom, boom, boom. In the side streets, you have to slow down because there's houses all along these areas. So the surface area here is going to be your little houses, right? Your little tissue cells. And so we have to slow things down here. Then we go back to the bigger vessels, so your vena cavas, for example, and now the velocity of the blood flow increases because we're back on the freeway. So that's an easy way of remembering that. Then autoregulation, all this refers to is that locally our vessels and our capillary beds are able to locally regulate, do we need to dilate, do we need to constrict? And this can be independent of the mean arterial pressure. And so what this does is that it's done to be able within the actual organs and the tissues, right, in our cells, to still maintain some blood flow. So say when the blood is shunted away from the gut, right? It's not like 100% of the blood is taken away or 100% is taken away from the kidneys. What happens is you remove a good percentage of the blood flow from those organs when you go into a sympathetic state, but the vessels themselves, for the blood that we do have coming in, the local vasodilation that takes place where there's that greater surface area helps still maintain blood flow going to the tissues. And so we're able to regulate that. Vasodilation of your arterioles, remember where we're going to have that relaxation of the precapillary sphincters where you're getting more blood. So this would be a metabolic, basically think of it as a chemical control. If you get more carbon dioxide, that means that oxygen levels are actually declining, right? Because now we're using up more of that oxygen. So now you're producing more hydrogen ion from the metabolic waste. And more potassium in some cases. Adenosine is also a local vasodilator that would cause this, and prostaglandins. And this adenosine that we're talking about here is not the same adenosine that we're talking about that directly impacts the SA node and the AV node. All right. So when you have the relaxation of the vascular smooth muscle, there's this release of nitrous oxide, which is a local vasodilator that happens from the vascular endothelial cells, and that also contributes to causing that vasodilation locally. All right. When there's vasoconstriction, remember that what happens is that now you're having a sympathetic response, and then you could have an increased constriction that would occur in some of these regions. But primarily what is happening, remember, vasoconstriction is occurring back in those larger vessels, and then locally, where we are local, we need more of that dilation to still maintain the blood flowing to the tissue. Then the vascular muscle also is kind of cool. Now we have a myogenic control. So what this means is that when blood enters into a vessel and causes pressure and dilates it, so it pushes against the walls, you give this thing called passive stretch. So now you have this increased intravascular pressure because of the volume, and what it does is that the muscle itself will actually want to return to its original length. So it's like you stretched it a little bit, and then it kind of constricts a little bit. So what that does is it creates and adds to the pressure gradient. If what happens, you lose more blood flow through that area and there's decreased blood flow, then what's going to happen is you're going to be like, wait, I need a little bit more blood coming in here. And so now what it'll do is the actual reduced stretch will cause the vasodilation to happen, and then we will have more blood going to the area. And then we get our pressure gradient back because that pressure pushes against the wall, returns back to its normal tone, right? And we get a little bit of vasoconstriction, and now we have our pressure gradient. So it's kind of cool how it works. And then all this is just a summary of what vessels will do. So green means these things are going to make the vessels dilate, and red means they're going to make the vessels constrict. So decreasing pH means it's getting more acidic. We're going to get more vasodilation. Decreasing oxygen levels, we're going to get vasodilation. This is a little decrease arrow. Then if you get increased amounts of carbon dioxide, potassium, prostaglandins, adenosine, and nitric oxide, that means this is going to cause dilation. And then within the vessel, in terms of stretch, right, or if there's any kind of damage or anything when you have vasospasm, this would cause a vasoconstriction. Then the sympathetic system, remember, is a constrictor. If you hit the alpha receptors with epinephrine and norepinephrine, they're going to cause vasoconstriction. Beta receptors, remember, cause vasodilation. And how it impacts vessels is that within our actual heart, remember, our blood vessels in our heart are arterioles feeding the heart muscle. They have locally within the vessels themselves some beta-2 receptors that respond to dilation. So when norepinephrine and epinephrine hit, the beta receptors in the blood vessels of the heart, they're called beta-2s just like the ones we have in our lungs, they actually cause within the heart for the vessels to dilate so the heart muscle can get more blood. Then angiotensin II, remember this is a very strong vasoconstrictor. ADH is a very strong vasoconstrictor. And then AMP is a vasodilator. Then in terms of blood flow and where it's going, right, when you're in a sympathetic response, blood is going to get shunted away from the kidneys and the abdomen. You're going to get a lot more blood going to your skeletal muscle, and you get more blood going to your skin because, again, you're going to be creating more heat. Going to the skin is going to help increase dissipation of heat. And then remember, you're also going to be sweating because of the sympathetic response going to the actual sweat glands. Then when you go back to a normal response, right, then blood obviously is going to go back to normal. But we actually have those temperature receptors in our hypothalamus that also help us with that in terms of regulating what the blood flow is going to do. So even if you are, say you're getting really, really hot, you're outside, you're not really doing anything, but it's just the temperature itself is causing you to get overheated, what will happen is you will have more of a sympathetic response because of that increase in body temperature, and then you'll get more blood flow going to your skin. But it's not, doesn't necessarily mean that you're going to get all this extra blood going to the skeletal muscle, but it will flow more to the actual skin to try and cool you off. Then when you have your increase in temperature, remember vasodilation helps dissipate some of that heat from the skin, and the heat will radiate out. And then the decrease in temperature, that is when you're getting colder, right? Now you're going to have that increase in vasoconstriction, so you'll have more vasoconstriction in the vessels that go to the skin, and then more blood will be shunted deeper because now we're trying to maintain and protect the vital organs. So we get more blood taken from the skin to the core when you get cold. And then long-term autoregulation, what this refers to when you see the word angiogenesis, in our hearts and in our muscles, skeletal muscles that are more aerobic, what will happen is when you're doing it healthy, right, when you're doing cardio, your body will adapt to what you're doing and increase capillarization. So you actually get more little blood vessels going to the tissues that are requiring more oxygen because you are more metabolically active and you need more oxygen going so you can continue to make that ATP. But what goes wrong when this happens is when you have a blockage or the vessel is starting to narrow because of plaque buildup or whatever, and now you have little vessels that are growing around the blockage so that the tissues still can get oxygenation and blood. And what happens is over time, there will be a point where these little vessels can no longer keep up with what the main vessel was delivering. And so that's when you start having problems and having the symptoms of chest pain or having peripheral vascular disease, for example, in your legs, where you now have a lot of pain when you're even just walking a little bit or going upstairs. So that's when angiogenesis is a bad thing, because this is when you have an occlusion going on. Then back to what we talked about in the beginning with the central circulation and how you have your heart that pumps the blood. And then we have in the periphery the blood vessels that contain the blood, which is our fluid. So when you go into shock, what can happen is central circulation shock is classified under cardiogenic and obstructive, and then peripheral circulation shock is classified under hypovolemic and distributive. And then under distributive, we have the anaphylactic, septic, and neurogenic. So remember, if you get a question on what type of shock is anaphylaxis, it's distributive shock. If you get a question on what type of shock is septic shock, it's distributive shock. All right, but it's all tying into what happens in the periphery. So when you have cardiogenic shock, it's the cardiac pump failure. The heart cannot circulate enough blood to maintain adequate periphery, which means you're probably having an MI, right? Heart cells are dying, or there's a conduction issue that is now affecting when the heart is going to contract and the blood is not able to leave, right? So the pump is being affected, or you have an arrhythmia that's affecting the pump. So that's a cardiogenic shock. Obstructive shock is going to be when blood flow becomes blocked in those vessels of the heart, or in the aorta, or the pulmonary trunk, or in those that are leading to the actual lungs. So causes of obstructive shock could be an aortic dissection, an embolism, right, a PE, or a cardiovascular vascular embolism in the cardiac arteries, or what could also be happening within the cardiac tamponade when the actual heart itself filling is decreased because there's some kind of pressure on there, or you could have a tumor within the ventricles or the atria that is obstructing blood flow. So anything that obstructs blood flow in the heart or those immediate vessels coming off is called obstructive shock. Hypovolemic shock, we're talking about volume loss. So hemorrhage, obviously, external bleeding, or you have internal bleeding. You could also have a plasma fluid shift that happens where the fluid is now shifting out of the vessels. Now this plasma fluid shift often happens when you have a distributive shock situation. So it'll actually end up leading to hypovolemic shock. So with anaphylaxis, remember what happens here is you have this extreme histamine release that is mediated by IgE and those basophils that are secreting a ton of histamine. So now what happens is you get this widespread vasodilation, and the edema that results from the widespread vasodilation, specifically up in the throat, can then cause that occlusion of the tissues in the throat and then cause the person to not be able to breathe. So that's your anaphylactic, right? The rescue for this is going to be epinephrine. Then the septic shock, what happens here is now again, you have widespread infection, but now that causes widespread vasodilation because it's trying to get everywhere where the infection is. And this widespread vasodilation is going to decrease cardiac output because it directly impacts venous return and coming back. And it can also cause multiple organ dysfunction syndrome, which we will talk about later when we get to more of the areas about blood flow. And then the neurogenic shock, this is what happens with spinal cord injury, where wherever the spinal cord injury has occurred, below that injury, remember, spinal cord contains the postganglionic or the preganglionic neurons that are going to go to the postganglionic neuron that then innervates the actual organ. So with neurogenic shock, one of the things that you would notice is that, for example, a person is not sweating below the spot where the injury is. Because remember, sympathetic response is what you're affecting here with your neurogenic shock. And so vessels below that would be vasodilated. Sweat glands below that would not be stimulated because you've now actually affected sympathetic function. So it all comes down to our pump, right? And any shock is going to affect preload. If preload is affected, then contractility is going to be affected in the heart. Then if contractility and preload are affected, then obviously what's going to be impacted is going to be what happens after. So afterload will decrease when preload goes down because you have decreased the amount of blood that's in the vessel, and if you don't have enough tension, say you have a neurogenic shock, that would actually impact afterload. And then the amount of blood that we have is going to directly impact preload, because remember, preload is dependent on how much blood returned to the heart and the stretch that took place in the heart. And then contractility is impacted because when the heart stretches, it can contract more forcefully. So if you decrease preload, you decrease venous return, it's going to decrease contractility. And then when it's right-sided heart failure, so primarily this is an easy way of remembering it, what it's going to do, it's going to have a problem in terms of end diastolic volume. So you're going to have less, right? Because what's happening here is now that right side of the heart is actually not emptying the way that it needs to. So when this side doesn't empty properly, that means that it's already going to have more end diastolic volume here. And so what happens is as that venous return is coming back, you're going to get backflow, all right? And so then in the end, what ends up happening is this end diastolic volume ultimately is going to be affected because now you have this right-sided heart failure. Just think about it. If this heart is not emptying properly, right, it's going to the lungs, then venous return comes back to the left, and then this gets pumped out. So if this is weak on the right side, you're not going to pump as much blood to the lungs. Not as much blood is going to return to the left ventricle, which means not as much blood is going to get pumped out into the systemic circulation. When that blood that gets pumped out into the systemic circulation is less, then that in turn will eventually end up affecting how much returns, and now you're going to get less EDV. And so increased ESV or less EDV are both going to end up causing problems with that peripheral edema. On the left side, now if you have left-sided heart failure, now the left ventricle is weak, which means now it can't pump as much blood out into the periphery. And so now that also will end up impacting what happens when the blood returns back to the heart if you're not pumping out as much blood. So that would then end up causing less end diastolic volume, right? And then what also happens is if this isn't emptying properly, now what we have is an increase in ESV with a weak heart. And as that increase in ESV continues, now the fluid returning back to the heart will start backing up into the lungs, and then that is why you end up with pulmonary edema, and the patient is having issues with fluid building up in their lungs. So right-sided heart failure is going to lead to peripheral edema, and then left-sided heart failure will end up causing problems with pulmonary edema. Eventually, they'll both cause issues. And then cerebral perfusion pressure is a very important concept because this is what we need in order for our brain to be perfused. Normal for our cerebral perfusion pressure is 50 to 1, or normal is 70 to 90. Extremes would be obviously 50 to 150, but we can deal with that, okay? But remember, this is going to be a function of your mean arterial pressure and what is happening inside the brain, right inside that cranial vault, your intracranial pressure. So this intracranial pressure directly is impacted by fluid in the brain, not enough blood flow, too little blood flow, too much blood flow. All of that is going to end up affecting it. But what ends up occurring is that your cerebral perfusion pressure is directly dependent on these two, your MAP and your ICP. Your ICP needs to be less than your MAP, because if the ICP goes up, it's going to directly impact mean arterial pressure, and it's going to decrease mean arterial pressure, right? So if this goes up, this here is going to be impacted, and then this is going to be impacted. And so what you don't want happening is for your intracranial pressure going up so much that it ends up impacting MAP, which then decreases the cerebral perfusion pressure. So adequate pressure, right? Between 50 to 150. This is when normal autoregulation, our brain can deal with it, okay? So when you have your cerebral perfusion pressure, that's this right here, okay? And this is how much blood is flowing into the brain. So if for whatever reason, all of a sudden, your brain here, right? We start increasing our cerebral pressure, right? We continue to increase here. So dilation here, right? Getting too much dilation would obviously be arterial collapse. So if you end up with way too much generalized vasodilation, it's going to directly impact what happens here with cerebral perfusion pressure and obviously your blood flow. So when we have autoregulation working, we can deal with that, all right? So what happens here is, suppose that your blood flow is being decreased, right? We need more oxygen, okay? But we can deal with that. Blood flow decreases, we need more oxygen, so what the body does is it's actually going to cause an increase, right? You're going to start dilating right here if that cerebral pressure starts to decrease and the volume. Now, as your pressure continues to increase, right? This is your normal regulation here. That's okay. We can deal with it, right? We're not going to bust open and have all this increased significant blood flow that's going into your brain. But what will happen when you get to the extreme, and this is when you get to your hypertensive crisis, all right? Our autoregulation fails. And so because you have so much pump pressure coming in that is increasing the blood flow, right? Because remember, your blood flow is going to increase if you increase the pump pressure. Now what's going to happen is the vessels are going to be like, whoa, we need to increase the dilation because we have so much coming in. And so then what the arteries do is they backfire and they start actually dilating and dilating and dilating and opening up to that point where up here in the brain, you end up having a significant increase in blood flow and pressure. And now you're going to get an increase in hydrostatic pressure. And that is going to start pushing fluid out into that cranial vault. And now you're going to get fluid outside the brain, pushing down on the brain. That increases your ICP, and then that is going to directly end up impacting what happens with the MAP, right? Initially, the MAP will try and fix it, but eventually the ICP will be up so high that then what's going to happen? It's going to end up affecting your cerebral perfusion pressure because now you're losing fluid, all right? And so that is one of the big risks of what ends up occurring. So during normal, our body can deal. But then when you start getting up into bigger problems, initially we'll have some of that vasoconstriction, but then what happens is you get this forced dilation, and then boom, you're going to get way too much fluid going into the brain. So if ICP increases, cerebral perfusion pressure is going to decrease, because if you think about it, think of this as the vessels in the brain are actually being sat on, right, by the increased ICP. The MAP is going to try and respond by increasing initially to try and maintain the cerebral perfusion pressure. But then what's going to happen is that continuing widening pulse pressure, so what's going to happen is systolic continues to go up, diastolic is going to start to drop, and the ICP will continue to increase, and then eventually, because now you could start having herniation of the brain, so you have that medulla oblongata starting to push through the foramen magnum and the occipital bone, that then is going to directly start impacting what happens in the heart, and now you start having bradycardia. So you're going to have widening pulse pressure, bradycardia, and the respiratory rate is going to be really irregular and weird. And so this is called Cushing's syndrome. Widening pulse pressure, bradycardia, and irregular respiratory rate. Then CO2 levels, as you know, will increase in the brain, because now what's going to happen is we don't have a means for our brain to get the good oxygen that's supposed to be flowing in there, because you're basically squishing your brain. And so now what's going to occur is acidosis, and now we get hypercapnia. And the hypercapnia, remember what our vessels do, because we have chemical vasodilators, right? Now they want to open up and dilate the vessel and try and fix the problem. And so then that will continue to increase the blood flow into the brain because it's trying to get more blood, because that's what it's craving. And then the ICP continues to go up, and then that directly continues to impact CPP, and then basically you end up squishing the brain so much that the blood flow stops and you die. And so that's what we don't want to happen. And that's why these brain injuries are so critical. So if you suspect that somebody could potentially have a brain injury, right? A fall, they get hit with a bat, they fell but they're not sure if they hit their head or not. I mean, those are the kinds of things that you're like, I want to be safe, right? Just get them to the hospital and have them get a CAT scan done. But this wouldn't be one that I would do a high-risk refusal on. All right, so then I talked about ICP. So ICP decreases, vessels will dilate, and then remember that you need to have a continuous perfusion pressure or cerebral perfusion pressure of 50 to maintain blood flow normal. And so as it continues to start, as the ICP goes up and CPP goes down, the brain will increase the extraction of oxygen. And then what's then going to end up happening is that now our systemic, right, we're also abnormal in terms of how we're breathing. And so now our oxygen concentration is going to keep decreasing. And now our brain is going like, hey, I'm just going to dilate as much as I can and increase as much as I can because it needs oxygen. And then, of course, I talked about the MAP trying to respond and trying to maintain CPP. And then it just ends up crashing, and it's a bad thing. So ideally, what you want to do to maintain a MAP that is going to continue to oxygenate the brain, right, is you want an ideal 110 to 120 of systolic pressure. This get to 90 is not going to be enough, all right? But as you guys go through and you start learning about, hyperventilation and what happens with hyperventilation and, with cerebral injuries, the ideal is you want to maintain pressure, right? They always say you want to get to 90 or above. But from the research, what is shown is that the ideal systemic pressure that you want, especially for systolic, is 110 to 120 in order to maintain that brain perfusion. When you hypoventilate, again, that's going to cause hypercapnia, right? Because you're not breathing often enough. Now what's going to happen is carbon dioxide levels are going to increase. Again, that will cause vasodilation. Then if you get too much, obviously it's going to cause problems in the ICP, because now what we're going to do is we're going to increase blood flow to increase ICP, because now we don't have the right amount of oxygen that's going, right? And now when you get this increased blood flow, that's going to continue to increase ICP, because now what is happening is you get more blood, more fluid, more hydrostatic pressure, and that is going to continue to push fluid out. Then, of course, the MAP responds because of what's happening and try and maintain CPP, but as you squish the brain with the increased ICP, that will then eventually end up affecting cerebral perfusion pressure, and then, right, and then the brain doesn't get oxygenated. Hyperventilation, this is hypocapnia. So remember, this is going to be your partial pressure of carbon dioxide is less. What this does is it actually results in vasoconstriction. So vasoconstriction will take place in the brain, and now you get less blood flow, right? Less oxygen. We retain carbon dioxide, but the decreased blood flow and volume are going to then impact what is going to happen. So now what happens is, like you'll read sometimes, it'll say you need to hyperventilate for a person that you're suspecting that's having a brain herniation or increased cranial pressure is going up. But it's not like you're having to hyperventilate up to like 40 breaths per minute, right? We're looking at like 20, 22. It's not a significant hyperventilation. And so what you want to do is, because what you don't want to end up impacting is that severe vasoconstriction, because then that will directly impact cerebral perfusion. You have less blood flow when there's cerebral vasoconstriction. So the perfusion is affected because you have less decreasing oxygen. Sorry about that little choking incident. I'm fine. So what happens is you end up getting less oxygen going to the brain because you have less perfusion, and then that can lead to brain ischemia, which means not enough perfusion, decreased oxygen. Now the brain suffers from not receiving enough. So what is happening here on the right side, here you see what happens when CO2 levels continue to increase. And so what will occur is that as CO2 levels go up, that also means that your partial pressure of oxygen is actually decreasing. And so what's going to happen eventually is that you're going to continue to get this autoregulation of vasoconstriction taking place. And so the higher the CO2 levels get, the more constriction that occurs, and then obviously that's going to end up affecting what happens in terms of perfusion in the brain, because now blood flow is going to continue to decrease the higher the CO2 levels go up. Then what you don't want to happen, obviously, is then all of a sudden you'll get to that point where, now we don't have enough. So then MAP is going to take over, and when MAP takes over, now you have this huge up going up, and now we try to vasodilate those vessels, but now the problem that can occur is then we end up with intracranial pressure and too much fluid leaving the brain, and then it ends up impacting the actual tissue, and then it ends up affecting that. So too much constriction, too much dilation, that is not where we want to be, and then that's what ends up affecting the actual issues with the actual brain. And then in our embryos, I just wanted to real quick touch on what happens in embryonic development. So we have a heart right about week 4, and it's going to be pumping and everything by about week 7. And what happens is that we're going to have adaptations that occur within the actual circulatory system of the fetus. So what we have is our placenta, right? And this is the filter, right? So mom's blood, baby's blood does not mix because we have a placenta. But the oxygen, the nutrients, the stuff that the baby needs are going to be passed through into the placenta. And then the blood, the good stuff, oxygenated blood, is going to pass in to the baby through the actual umbilical vein. At the umbilical vein, we have an adaptation, right? We're going to have a distribution of some blood going to go in the liver. The rest is going to go up into the inferior vena cava. Then from here, the blood will go into the right atrium, down into the right ventricle, and also from the right atrium, some blood will shunt to the left ventricle through another adaptation that's in between that's called the foramen ovale. That will actually help bypass some of the blood that would normally go down here into the right ventricle, because remember, this right ventricle is going to pump to the lungs. But we have another adaptation that has to take place because these lungs are in fluid. So the lungs, the right ventricle will pump into that pulmonary trunk, and then right at that portion where the split is to go to the pulmonary arteries, what happens is we have another adaptation called the ductus arteriosus. And so this blood that's being passed in that would normally go to the lungs will now get shunted over to the aorta, and then that will get distributed through to the baby. So our 3 adaptations that we have are, we have our umbilical cord, obviously. This one is going to carry oxygen-rich blood, and you only have one umbilical vein that carries the oxygen-rich blood. And then we have the umbilical arteries that take the waste and the carbon dioxide-rich blood back to the fetus and the placenta. So again, your umbilical vein comes in. First adaptation is the ductus venosus, and it's at the level of the liver and the inferior vena cava. Then that blood will continue up, meet up with that right atrium, where you're going to, the baby's blood basically from the head and from the lower body, right, are coming into the right atrium. That blood will enter the right atrium. A good portion of it is going to get shunted across to the left atrium. This left atrium will then obviously bring the blood down to the left ventricle, and when that left ventricle pumps, it'll pump into the aorta and distribute the blood. The other adaptation that's going to happen is after that foramen ovale, we need that second adaptation called the ductus arteriosus to bypass the lungs. And so the blood that goes from the right ventricle into the pulmonary trunk will not go to those lungs. It's going to get bypassed directly into the aorta through this ductus arteriosus. And then that blood will circulate and get distributed through into the organs as needed, right, through the aorta. Then the blood that passes down into the common iliac arteries, the blood that goes into the internal iliac arteries, this is where the umbilical arteries are connected. And so from the internal iliac arteries, the umbilical arteries will then return the blood back to the placenta and then do the gas exchange. So the baby itself, its hemoglobin is actually different than the hemoglobin we have outside of the womb. And it's able to transport the oxygen in the manner that the baby needs. And it's able to also carry the oxygen and unload it in a manner that is much more efficient than what we have right now. So once you're outside the body, right, we have air. But in the fluid environment, the hemoglobin has to be able to carry the oxygen properly and then oxygenate the tissues that are needed and are requiring the oxygen, because obviously baby's growing, baby is developing, fetus is going through the various transformations that it goes through to become a little human baby. And so the tissues have to have oxygen, right? But this is how we're able to do that adaptation. So at the liver level, we have the ductus venosus, where the majority of the blood is going to bypass the liver, enter the inferior vena cava, bring the blood to the right atrium. Then, to bypass the lungs, we're going to have the foramen ovale between the right and left atrium, so that shunts the blood across, and then the ductus arteriosus will shunt the blood from the pulmonary trunk, pulmonary arteries, into the aorta. And so when the baby takes its first big breath at birth, this foraminal valley kind of like gets suctioned shut, but the tissue itself that closes the opening, that takes about a year to close. Same thing, these other little adaptations will, when the baby takes that big deep breath and there's that big change in pressure in terms of what happens with the fluid environment, they start to close off. And so it's going to take some time for the actual tissue, which is connective tissue, that is going to form between these openings and then completely close up. And so you guys will be learning as you go through resuscitation and everything what to look for if this foraminal valley hasn't closed properly. And obviously this will be more pre-hospital stuff that you're learning, so it's not like you're going to be in the NICU and being the NICU team that is going to be taking care of all of this. But these are 3 adaptations that you do need to know: ductus venosus, foraminal valley, ductus arteriosus, and then also remember that the umbilical vein is what carries oxygen-rich blood to the baby, and the umbilical arteries are what take the blood away. And that is the end. So I'm sorry for the coughing, alarms, different things that happened throughout the lecture, but just wanted to power through this and make sure that we got through it. Again, if you have any questions, please let me know. This chapter is probably one of your most important ones in terms of getting a good foundation for what is taking place. And probably went a little bit over with what I talked about, the brain and brain perfusion and that. But please make sure that if you have any questions on that, let me know. And don't forget to do the study guide. That is where I've focused your studying for what's going to be coming up on the exam for the cardiac stuff. And then we will move on now to the next chapter.